so much for joining our symposium about translocation. I'm actually really thrilled with the number of experts that are willing to share their knowledge and experience. I'm pretty sure that there's a talk for everyone. Personally, I'm pretty curious about what socks have to do with mycorrhizal fungi associations. I'm sure I'll find out. Now, this symposium is prepared by the Australian Network for Plant Conservation and funded by the Ross Trust, which means that it'll be, it's free for you to attend and it'll be stored on YouTube as well. My name is Chantelle Doyle. I'm the project manager of Translocation for the Australian Network for Plant Conservation, and I'm also doing a PhD in translocation. I just participated in the first ever International Plant Translocation Conference, and I am pleased to say that Australia is really up there in terms of expertise and experience, and also that this is a practice that undoubtedly is going to keep growing. We're going to be doing something a little bit different this time. We're not going to be having set time for questions. Instead, we would invite you to type all of your questions for any of the presenters in the Q&A section, and then the presenters will answer those virtually. You can also contact us if you didn't get something answered, and we'll forward on the questions. If there's a little bit of time at the end of the session, we may have some questions, but we don't want to guarantee that. If you are experiencing any problems, please let us know in the chat. Jo Lynch is sitting there in the background and she'll be moderating the chat. Also, you can contact Christine Finance, who is running Zoom in the background. She's going to be working with the panelists and the presenters on the email address that'll be listed in the chat as well. So our first session is going to be about context. First up, we're going to have Dr. Lucy Commander and Jen Silcock. They're going to be setting the scene. Lucy is giving us a snapshot of the Translocation Guidelines. Yes, Australia has some. And Jen is going to be giving us an overview of the status in translocation in Australia. And I'm tacking on a tiny bit to the end of that as well. Then we are very fortunate to have Neville Walsh from the Royal Botanic Gardens. He's the senior botanist there with years of experience, and he's going to be talking about how the ecology of plants can impact their establishment for translocations. Then Michael Longmore from Merri Creek Management Committee is going to give us a practical example of plant ecology and how long it can practically take to crack the secrets of species so they survive and recruit. And to end our first session, Dr. Megan Hirst from the Royal Botanic Gardens is going to give a little practical guide for optimizing seed collection, processing and storage, so you can maximize any end use, including translocation. Now, I'm going to sort of disappear for a little while and join you back at the end of the session.
Hi everyone, my name is Lucy Commander and I was the ANPC project manager for the third edition of the guidelines for the translocation of threatened plants in Australia. There are a lot of threatened plants in Australia, but there are also a lot of conservation actions that we can use to prevent extinction of this threatened flora. One of these is translocation. Other options are habitat protection, threat management, as well as ex situ conservation. So today I'll be talking about translocation. Translocation is the intentional transfer of plants or regenerative material from an ex situ collection or natural population to a new location. Now, there are different types of translocation depending on where the plants are sourced from and where the recipient site is. So an introduction is an establishment of population in a site that has not been, where the species has not previously occurred, but it's within the known range of the species. Then there's reintroductions, so that's re-establishing populations in places where it no longer occurs or it's become locally extinct. Reinforcement or augmentation is about adding individuals of a species into an existing population, and assisted migration or assisted colonisation is where we aim to establish a species for a purpose of conservation outside its indigenous range. Because there have been increasing translocations since 2004, this has led to more information about translocations, so we're able to do them better. But it's also increased the need for more information. And best practice guidelines such as these translocation guidelines can help share information. ANPC published translocation guidelines in 1997. That's when the first edition came out, and then the second edition was published in 2004. So I worked on the third edition, which was published in 2018. These guidelines are widely cited by conservation agencies and those who are underdoing translocation. So for our revision process, firstly, the funding was granted from the National Environmental Science Programme's Threatened Species Recovery Hub under Dave Coates' project. Then the ANPC formed a steering committee consisting of Dave Coates, Linda, Bob, Kathy and Maria. They appointed a project manager, which was me, and we found authors and hosted a workshop to discuss the content of the third edition. We had over 30 chapter authors and case study authors. We started with an initial workshop and information day in Sydney, and we also had a genetic mixing workshop in Melbourne. Then we wrote, edited, and did the layout, and finally launched. The content of the Translocation Guidelines is essentially in three components: planning, preparation and implementation, and monitoring and evaluation. The planning phase consists of an introduction, a chapter on decision making, so deciding whether or not to translocate, a pre-translocation assessment of biology and ecology, selecting resource and recipient site, as well as policy approvals and translocation proposals. Then we move on to pre-translocation preparation in Chapter 6 and in Chapter 7, implementing the translocation and ongoing maintenance. Finally, there's a chapter on translocation monitoring and evaluation. The last chapter is really all-encompassing and refers back to all of the previous sections, and this is about community participation and support. So now I'll go through each chapter in a little more detail. Chapter 2, deciding whether or not to translocate, consists of information such as conservation actions available, the benefits and risks of translocation, deciding whether or not it's necessary, setting goals and objectives, and it also has a decision-making framework. Chapter 3, the pre-translocation assessment of biology and ecology, talks about the importance of recovery teams and translocation working groups, which bring together people from a range of disciplines who have experience in conserving a species. It talks about the need to collect all the information about the species that's needed for the translocation, such as the biotic interactions, so for example, what are its pollinators, how to source the plants, whether you should collect seeds or cuttings. And all of this information informs everything from site selection to monitoring. The information then goes into the translocation proposal. This is a template that we've drawn up for a translocation proposal. Some states and territories have their own translocation proposal which must be used, and other states and territories don't have a mandated proposal template, in which case you can use the one in the guidelines. Chapter 4 is on selecting source and recipient sites. It talks about how to select a source population or source populations, and whether or not you should mix these source populations, and how many individuals to collect from. In terms of selecting recipient sites, this can be done in a way to decrease the extinction risk, and you need to think about how many sites and undertake a site assessment to make sure that it's suitable. When selecting sites beyond the known range, known as assisted migration, you have to make sure that the abiotic and biotic components are suitable. For example, is the soil type correct? Are there pollinators? And there's a potential use for predictive tools to be able to do this. The chapter also talks about preventing unintended consequences of translocation. Chapter 5 is on policy approvals and translocation proposals. Translocation policies explain or guide how a government, authority or agency will process or consider proposals to translocate, and there might be other policies that are relevant, such as those for threatened species. Different jurisdictions have different requirements for approvals, so there might be licenses and permits required, and you might need to have multiple approvals from different authorities. Ensure you have consent from traditional owners, and all of these approvals can be quite time-consuming, so make sure that you start applying early. Chapter 6 is on pre-translocation preparation. It talks about the people and resources that are required, the timelines that it might take for this preparation phase, how to collect material, how to undertake ex situ collections, experimental design, and also planning for management, monitoring and evaluation, as well as site preparation. Then Chapter 7 is about implementing the translocation as well as ongoing maintenance, and it has information about the timing of translocation and whether or not it might be related to rainfall, ensuring the plant age and health is appropriate, as well as acclimation. It goes through how to prepare plants, how to plant, and also some options for after planting care, such as watering, plant guards, and other site management. Chapter 8, Translocation Monitoring and Evaluation, talks about documenting what you're trying to achieve with these goals and objectives. It explains how to give yourself the best chance to explain the results by using explanatory variables, which could be management or the environment. It talks about how you can permanently tag and census all plants, and it talks also about monitoring frequency. Perhaps you may like to monitor frequently in the first year and then annually. Be clear on what you're measuring, but also look out for surprises. The final chapter, Chapter 9, is on community participation and support. So the community around the translocation could be NRM bodies, local governments, consultants, perhaps the nursery industry or nurseries that have been growing the plants, friends of groups, traditional owners, native plant societies such as a wildflower society or an orchid society, as well as local environmental centres. Ensure you engage with the traditional owners in the co-design of the translocation process, especially recipient sites, to consider the potential impacts on cultural values. Think about asking the community for practical support, such as assistance with surveys or reducing threats. And the community are also really good local contacts as well. They can help to raise the profile of the translocation or the particular threatened species that you're working on. And there's also an option for citizen science. And finally, when the translocation is complete, make sure you celebrate the achievements and recognise the community. The Translocation Guidelines also has some additional resources. In the guidelines themselves, there's 23 case studies, and they are abridged in the guidelines and they illustrate points within each chapter. But the full versions of these abridged case studies can be found in Australasian Plant Conservation, or APC, which is a bulletin of ANPC. And they're also found online at this website here. The species for these case studies include orchids, macadamia and Willamette pine. And there are also videos on our YouTube website. There are 2 other best practice guidelines which complement the information in the Translocation Guidelines. These are the Plant Germplasm Conservation in Australia, which are guidelines for ex situ collections, and they expand on the information in the Translocation Guidelines by providing additional information about genetic guidelines, seed and vegetative material collection, as well as seed banking and the plant nursery. The Flora Bank Guidelines are best practice guidelines for native seed collection and use, and while these focus on all species and not just threatened species, they go into much more detail about seed sourcing, collection, processing, as well as drying and storage and germination. You can find free copies of both of these guidelines on the website. Although these guidelines have only been published in 2018, we've achieved a significant number of page views and also distributed a number of print copies as well. There have been citations in the peer-reviewed literature, but more importantly, the guidelines have been integrated into state policies such as the New South Wales Translocation Operational Policy. They've been cited in national strategies such as the Threatened Species Strategy Action Plan, as well as recovery plans, conservation advices, translocation plans and proposals. So a number of key messages from the Translocation Guidelines. These guidelines are a how-to guide for threatened plant translocation. They really should be used by everybody in Australia who's undertaking threatened plant translocation. They're a useful way of disseminating information to end users as well as policymakers. They're written in plain English, so they're very easy to understand for students and also to researchers who are new to the field, as well as friends groups and other community members. They've been a great way to involve all parts of the sector. They're free to download as well as inexpensive to purchase, And much of the information inside the guidelines is applicable to common plant species too. So I encourage you to download the guidelines today if you haven't already, or purchase a print copy, and make sure that you use it when you're next doing a translocation. Thank you so much to all of the authors of the Translocation Guidelines. I really appreciated your input and support during the publication of these. I'd also like to thank all of the organisations that provided funding and in-kind support, as well as all of the committee members of the Australian Network for Plant Conservation. Thank you.
So this is a talk I gave a few years ago at the ANPC conference, updated with a couple of details, but basically talking about Australian plant translocations over the past 60,000 years. I guess when we started compiling the Australian Translocation database, which was back in 2016 as part of a NESP project led by the West Australian government and the Uni of Queensland, I realised that despite a lot of literature about translocation being a new and emerging sort of science, which it is in a lot of ways, it has been occurring across the world for at least millennia. And it has included a range of plants, food, medicinal, and even narcotic plants, often with cultural or ceremonial elements. And that's quite well documented for lots of plants across the world. In recent years, there's been a lot of public interest in books about Aboriginal environmental impacts and environmental management in Australia, particularly Bill Gamage's book and Bruce Pascoe's Dark Emu, which a lot of people, even non-scientists, have read and are really engaging in trying to understand and learn about this. So that's one thing we still don't know is how widespread this intentional dispersal and cultivation of plants was in Aboriginal Australia. And when I was compiling the translocation database about modern translocations, I also started looking into some of the archaeological, ethnographic, biogeographic, and increasingly the sort of phylogenetic approaches to look at ancient plant translocations in Australia. So what I found was that more than 50 species are recorded as being deliberately translocated. So you remember Lucy's definition about plants that are propagules that are deliberately moved to somewhere else, either within or outside of their sort of current range. That involved mostly trees and shrubs, but also a lot of tuberous species. So that's the yams that occur right across Australia. It was mostly important food species and plants that were used for materials, but there was also at least 17 with some ceremonial or cultural importance. And with these ones, particularly in Central Australia, translocation often occurred as part of a specific ceremony, what we might broadly call increase ceremonies. Most of these translocations would fit into, with Lucy's definitions in the guidelines, introductions and reinforcement, and they often involved nurture of the plants and the habitats, so soil management and watering of the plants as the propagules were dispersed. 10 of the translocations were post-contact, so often in Central and Northern Australia, people have moved to towns and properties, outstations that may be some distance from their country or certainly from parts of their country, and they've moved plants and planted them in their gardens to maintain connection to these important sites and patches of country. Just quickly, this map shows the number of translocations per biogeographic region, number of ancient translocations per biogeographic region in Australia. You can see Central and Northern Australia have comparatively more documented. Possibly that reflects the fact that there's still people living, I guess, semi-traditional lifestyles, and anthropologists have worked with them, been in the bush with them and observed these practices happening. The translocations that are documented in the ethnographic literature would, I suspect, represent a tiny fraction of what was actually done. This has all been published in a 2018 paper in the Journal of Ethnobiology, if you want to look into it any further. Suffice to say for this forum that, yeah, there's many undocumented translocations, and there's also contemporary implications for the translocation of these ancient translocations for debates that we're having today about assisted migration. And it provides this real challenge for our conceptions of what are natural species distributions. And this field of research is sort of burgeoning in the fact, there's these new molecular and genomic techniques that have been used to tell these really neat stories about some plants like the Central Australian cabbage palm, the boab, and the black bean in the rainforests of Eastern Australia. And the signatures of ancient translocations are evident in those genetic signatures of the plants. So continuing along, but generally poorly documented in the literature, at least, history is the modern translocations. So when the NESP project started about 2016, there was a realization that there was increasing translocations, including for development mitigation. It's high risk and labor intensive. It also has a really low rate of publishing, and there was no real understanding of what had been done or where or how successful it had been. So the NESP project that was led by David Coates in Western Australia, and Laura Simmons and myself were behind assembling the database along with numerous researchers from Western Australia. We basically started a process of interviewing everybody we could find who'd been involved in translocations. It was a bit of a snowballing process. Someone had mentioned that someone else had done something. So we ended up talking to more than 150 translocation practitioners, either face-to-face or on the phone or via email. And we pulled together what we call the Australian Plant Translocation Database, essentially a big spreadsheet of more than 1,000 translocations of 376 taxa. And a version of that is now available online at the ANPC website via that link. So just a quick snapshot. This was published in a paper in Biological Conservation if you want further details. But you can see the increase of translocation over time, particularly those mitigation translocations, which occur when a population of a threatened species is going to be impacted by development, so urbanisation or road widening or construction or mining. The map down the bottom shows the number of translocations per bioregion in Australia. These are just the modern translocations that have occurred since about 1970. The red crosses are the development mitigation translocations, and the green stars are the conservation translocations. So you can really see the practices being concentrated right down the east coast and across southern Australia into southwestern Australia. We did have a look in the paper about evaluating the performance of the translocations. We had data to be able to do that for just over 700 translocations. Yeah, so quite a few of them had no plants surviving or fewer than 10 plants surviving. And yeah, fewer plants surviving than is considered necessary to establish self-sustaining populations did differ a little bit with habitat. Some rainforest plants, we relaxed that a bit, but it was generally 50 plants was our cutoff to avoid sort of genetic problems and be able to create a self-sustaining population. So that's basically over 60% of translocations that had not succeeded in creating a self-sustaining population, including 70% of the mitigation translocations. Of the 270 with more than 50 plants surviving, 2/3 had no recruitment into the populations, although that might have been too soon for some translocations, especially of long-lived shrubs and trees. There are only 93 translocations that had more than 50 plants and some recruitment, so indications that these might become self-sustaining populations. And Michael Whitehead and Peter Vesk, and Pete will be talking later in this workshop, but yeah, they are working on a paper at the moment looking at some more nuanced analysis of the translocation performance based on that database that we collected. So I guess the take-home message is you really need sufficient founder propagules. You want to be creating a population that's going to be genetically healthy and have a chance of recruitment. It was really difficult to predict translocation performance. It seems to be a real role for just inherent traits of species and habitats that interact with the factors that you can control around translocation treatments like watering and fencing. There's huge importance of long-term commitment and monitoring. And the main factor that once you were able to get survival of plants, the main factor that was inhibiting success was a lack of second generation recruitment. It's not just a problem for translocated populations. A lot of natural populations of these rare and threatened plants don't have any or extremely limited recruitment as well. The big factor seems to be some disturbances needed for a lot of these plants, whether that's burning or another form of disturbance to stimulate recruitment. I think you'd have to say at the end of this, translocation does remain largely experimental. So that sounds a note of caution for mitigation translocations in particular that may have destroyed or impacted on natural populations. It also highlights the value of a well-documented experimental approach and publishing the data if possible. That's it from me.
Thanks, Jen. I'm jumping back in just for a couple of minutes here because I'm piggybacking on Jen's presentation. I just want to talk about some work that I've been doing, just expanding on the database. So I'm just going to share my screen for 1 second. And while I'm doing that, I want you to get out your phones because I have a QR code for you to scan. Basically, off the back of Jen's database, or the database she led, I wanted to talk to practitioners and find out some of their experiences. So get some qualitative data. Originally, I did want to talk about legislation, but because we weren't able to secure any government employees to talk about legislation, there seems to be a little bit of a resistance, but we have reviewed the legislation in Australia and it's in that paper there if you want to have a look yourself. Bit of a dry topic for me to talk about, but you can look for your relevant state there. So basically, I've interviewed about 48 different practitioners from government, consultancy, community groups, researchers, even one developer anonymously had a chat with me. And I asked them a whole lot of questions that I'm happy to delve into off the record. But basically, I wanted to explore this that came up quite often, that mitigation and conservation translocations were perceived very differently, that basically people were a little bit skeptical about mitigation translocations, that they weren't sure about the success, that they thought they were a bit tick and flick. And part of the reason for that is because people couldn't find data, couldn't find information. I think this quote summarizes it quite well. I tried to find the reports that I knew were written, but no one seemed to know where in the government they went. This is an issue for all threatened species, that people who need the information don't get it so that they can learn. We can all learn. And based on that, I wanted to find out, well, how true this is. So along with Jen, we published earlier this year a little review of the 1001 records in the database, and actually found that 66% of the conservation translocations were unpublished, whereas 71% of mitigation were published. It's just that they were in grey literature, so people couldn't access them to find out information, which is exactly what that person was talking about. So I wanted to know, well, look, do you think that we should have a database that continues? Because the current database is a snapshot, and it stopped in 2018. And there was a lot of support, very broad support for an ongoing database. When I asked people if it should be compulsory, there was a little bit more reticence, mostly because all the work had already been done and they didn't want to have to replicate things. Also, some people had threatened species on private property and they didn't want to share that detail. But they did want to see data and information available, preferably at a national or even an international level. So this is the bit where I ask your opinion. If you could scan that QR code there for me and just answer the question, do you think that we should have a voluntary public registry? So not a database, because that can be quite onerous, but the public registry would just include the name of the species, a general location, who did it for contact purposes. So that would be either a person or a company, and why. And that would be it. Just to know if this is something that's worth pursuing and people think that it is worthwhile. Okay. That's me. I'm I'm going to duck off now and move on to our next speaker, which is Neville Walsh.
Right. Thanks, thanks, Sean. Thanks, Jen. And I'll put my hand up first of all to confess that I'm probably a criminal of doing a number of translocations ineffectually. But I guess if nothing else, it's made me realise the role of ecology, understanding the ecology of the species that we want to put out, is somewhat important, if not critical. Okay, so a lot of this stuff is absolutely obvious. The things that we consider before we do a translocation, and Lucy talked about those quite comprehensively. And you know, a lot of us will apply something like this matrix down the bottom right there, where you've got an increasing, for example, risk on one side and rarity on the other. It could be a whole lot of other things that are those axes. And obviously, the higher the risk and the higher the risk of extinction is what I'm meaning in this case, rather than risk of success. And of course, the redder the country that we get into, the more warranting translocation as any species might be. But it gets a bit more complex than that. You've got to start thinking of the likelihood of success. So we sort of introduce another dimension there, and we could start thinking about the ecological bang for buck. So what we actually insert into the surrounding vegetation, what values we insert into that vegetation by installing a species that is no longer there, or perhaps never was there, but we think it would do well to be there. So to state the bleeding obvious, you can read that intrinsic ecological requirements require extra knowledge about the recipient site and or various treatments for the plants to be located. And as I say, in my past, I have done translocations as scientific as throwing a few pots in the back of a ute and digging a hole and putting a plant in and walking away, which surprisingly has been successful in a few circumstances, but they're mainly on riparian sites. But that's anything but a deep ecological understanding of what's going on there. And I'm going to talk about a couple of projects that my colleague at the Botanic Gardens, Nushka Reiter, has worked on with some success, and particularly in orchids. And I know it's unfair to leap to orchids initially to talk about this stuff, because if anything has sort of boxed themselves into a bizarre ecological corner, it's orchids, and a species that's been long known to be on the skids in Victoria is Calodenia hastata, Melbourne's, or the Portland spider orchid. We've watched this thing decline over 30 or 40 years. And as you can see there, the current number of wild known mature individuals is fewer than 379, which is a curious statement, but I'll ask more about that. So surveys were undertaken at a whole range of sites where the plant had been known in the past, and particularly with a view to tracking down its pollinator. So the first step of this work, of course, is understanding just what the pollinator of this orchid is. Sometimes with orchids, they have generalist pollinators, and sometimes they have very specialized ones. And in this case, the pollinator is species specific. And curiously, I can't remember the name of the genus, but the species name of the pollinator, a little finned wasp, is also hastatus. Calodenia hastata is pollinated by something hastatus. Curious. So after all of those, lots and lots of surveys, 230 something sites there, the pollinator was found at only 2 of the extant sites of Calodenia hastata. but the pollinator was picked up at 5 sites that were either considered suitable for the species or sites that it had been known from in the past. So that guided the recipient sites really for this sort of thing. And of course, you know, growing orchids in itself requires an enormous understanding of their ecology, including extracting the fungus from it and growing the fungus separately. We all know the amazing stories of orchids. Anyway, so we got 448 plants cultivated. So already in the lab and the glass houses, we had more plants to propagate, to put out than were known in the wild. 2 sites were selected, and the sites that selected are quite often privately owned covenanted properties, and they are often the best sorts of sites because there's usually landowners who are very sympathetic and very interested in their orchids. And I should have preempted all of this by saying attempts at translocating orchids, particularly in the past, have been woefully poor. But in this case, choosing a site that had suitable vegetation and the pollinator was known, you've got 70% survival and 33% flowered and pollination and seed set. So, you know, all of the ingredients are there, and we're just sitting back waiting for germination of those seedlings in good years, which seems to be the case with these things. They're very dependent on good years for seedlings to be recruited. Now that was an orchid that's pollinated using sexual deception. So by producing a pheromone, the thinid wasp is enticed into attempting mating with the orchid, which is how the pollinia are transferred from one to the other. Caladinia versicolor, another spider orchid, was another target species for this behaviour. Known from three locations, but we know from herbarium specimens this thing was quite widespread across the Wimmera previously. And you know, a little bit about its habitat, of course. There's a couple of quite good populations. Nonetheless, fewer than 600 wild plants were known, or are known prior to this work. So you can see the yellow stars on that map indicate where it's known currently, and the green blocks indicate historical herbarium records where the plants are no longer known. And unlike the Caladinia hastata, interestingly, the pollinator for this thing isn't lured into the orchid by pheromones. It's actually lured in there for food. So this is a bee rather than a native bee, rather than a thinid wasp. Nonetheless, the insect is in there, attracted to the sugar, which is at the very base of the labellum, and in doing so, it's forced into a position where the pollinia are attached to the back of the insect itself. So these male bees and wasps, you know, searching for sex or food, that's a common story. The things that are used to make males compliant. Can I say that? No. Anyway, as this four covenanted properties were again selected for this work. You can see the fairly serious fencing that goes on here to keep grazers out of the sites. Natural pollination was found at all of the sites. 798 plants produced, a very high survival rate, which is fantastic really for a spider orchid. High degree of flowering in the first couple of years, and recruitment within 2 years of the planting, and then flowering and seeding of those recruits 3 years later. So through that action, through a deep understanding of that species ecology, doubled the wild, or the population out there of that particular orchid, which is a great result. Things other than orchids that have small critters associated with them, Grampian's globe pea Sphaerolobium acantus. Being a pea, of course, very commonly have nodulating rhizobia in their roots. And in this case, 3 different strains of rhizobia were tested for this species. They were extracted from the plant, but only one of those strains, if you like, were shown to be successful, and extracts were made of those, and they've been added to cutting grown plants, in this case of Sphaerolobium, to be translocated out back into the grampians, where, we talk about the intrinsic ecological requirements, the places where these things grow. Of course, they become part of the ecology, and part of the ecology out there, unfortunately, is replete with browsers, both native and introduced. So it's that follow-up maintenance monitoring that's going to lead to the hopefully successful translocation of this species. Sliding back to another orchid, in this case Thelymita epipactoides, threatened, listed in Australia, previously known from South Australia and Victoria, but now only in a few sites in South Australia and Victoria. It's a generalist, it's pollinated by generalist insects, so the story isn't so much about finding the right pollinator, but in this case, the mycorrhizal fungus that's associated with it, that's required to get the thing to germinate. 2 species, both of them new to science, both of them which subsequently have been named. And interestingly, although they'll both seeds will germinate in association with those fungi, they operate at different temperatures. So if you're wanting to translocate into a relatively warm site, you've got to make sure that you've got mycorrhiza A, and into a cooler site, mycorrhizal B. So this is getting to understand, the autoecology of these things, the intrinsic ecology of these plants, biology, deeply before we move on. A slightly more conventional plant, but with its own quirks, Borea mirabilis, the Grampian pincushion lily, the only southeastern member of this very interesting genus. It was known from one site in the Grampians and had never been shown to produce seed, and the reason for that appears to be pollen sterility. Nonetheless, we grew plants of this up, seedlings of this up, not seedlings, cuttings, cutting grown plants at the Botanic Gardens, and did a translocation site about 10 kilometres from the known site. Now that involved a rigorous matching of ecology, soil type, aspect, altitude, associated vegetation. And really, once you start trying to apply all of those variables that are known from the known site, the number of likely places really reduces very quickly in this case anyway. That does raise the question, though, when you're back down to a single population, do we know that that's always going to be at the optimal site? But it's the only thing we have to work on. Anyhow, the site was chosen, and look, it powered for 2 or 3 years, and there's a bit of a general patting at the backs. And I've got to say that to some extent the ball was dropped in terms of management, control, monitoring of those populations, and there's also a bit of track work in the Grampians, so that access to this site was no longer as simple as it once was. Anyway, after having not much done about that for a few years, subsequently this population was revisited and found that it was a good site for growing Borea, but unfortunately it was a good site also for growing the associated shrubs which had dominated the site, and that had been visited by, again, quite a lot of browsers. And that site didn't succeed. So the first part of it was good. The second part, the monitoring and follow-up, not so good in that case. We start thinking about the intrinsic ecology of some species and the likelihood of success of a translocation effort. Now, Astringia crenifila is a nutty plant, and it grows on these virtually sheer cliffs of the Snowy River Gorge along a single series of cliff lines, and it preferentially grows where there's no soil. So there's small cracks in the cliffs is where these plants hang out of, and I can only imagine that seeds are drawn into these cracks by ants. And anywhere where there's actual soil accumulation on a cliff line, the Astringia simply won't be there. You can see on that top shot there how this one of these little horizontal cracks is the home site for one of the plants, and really it stretches my imagination to think how we could successfully install plants into a site like this, or to a similar site. Now, to some extent, fortunately, the site is remote and reasonably secure from damaging creatures, but the site was burnt in those bad fires a few years back. Fortunately, there's so little vegetation on these cliffs. Maybe half of the plants were burnt, and some of them are bouncing back okay. So in that case, it's a situation where we assess the risk, we look at the likelihood of success, which in this case I'd say very low, and we choose to maybe keep plants going ex situ at some kind of insurance policy here. Interestingly, when you grow this species in a pot, it doesn't want to grow vertically, it wants to be on a cliff, so all of the plants just hang sideways out of the pot. But the great thing about this spot, really, that one of its saviours is the difficulty of access, is a bit of whitewater rafting to get there, which is nice. And now, I've talked about sort of, as I say, the autoecology of these plants, the things that are intrinsic to them, but sort of thinking about more broadly the ecology of these species, I just wanted to think a little bit about the ecology that plants actually add to the environment. So they create their own ecology to some extent. And the notion of keystone species here is pretty important. They're usually locally dominant, and they exert some kind of influence on the organisms around them. We think of things like Nothofagus forests of alpine sphagnum bogs and Western Plains grassland on the volcanic plain, and they have been the places where we have planted in threatened species. But I mean, I sort of think too that they might also be the species, these keystone species, where we might want to make new versions of them so that they can then become the recipient sites for those species. So the species may or may not themselves be rare, but the associated species might be. And maybe it's time to sort of start zooming in on some of these communities that these species create that currently aren't recognised. So you have dense stands of very rare things like Nematolepis frondosa from East Gippsland, dominant species in a very local area, and who knows what other species are associating with that. Similarly, a couple of grasses of the basalt plain, Poas helicostris and Poaphysoclina. A thing that I've been working on, associated with, is a recovery team for helmeted honeyeaters, and they're dependent on woodlands, dense woodlands of Eucalyptus camphora. So the limiting factor here is the number of worthwhile stands of Eucalyptus camphora. And I realise I'm running short of time, so I'm going to have to race through this. Again, big benchtop study involving particularly Matt White from DELP, really helpful in this program, looking at the number of sites where Eucalyptus camphora could be expected to survive, and then we go out there and look for birds just in case there is some there, they never were, and look for extant plants of Eucalyptus camphora. in very few cases there were, the conditions just right for re-establishing Eucalyptus camphora woodlands. But it turns out that there's in the one site where Helmet honeys occur, it's also the one site where lowland leadbeaters, possums are known to occur as well. So it's got these really strong drivers to such an extent that big properties, completely alienated properties, have been identified as sites that were suitable. So very, very long drawn out process, but a translocation of sorts, a habitat creation using Eucalyptus camphora. In this case, it's a place called Haining Farm on the Yarra River. It's near Alton. And we're hoping, in 10, 12 years, we'll have created habitat for helmeted honeyeaters. And I guess, as I say here, the moral here is you need financial support for Translocation Fund and the associated threatened animal. And I'm stopping. Thanks.
Thanks, Neville, and thanks, Sean. I'm Michael Longmore from Merri Creek Management Committee, and I'm going to be talking to you today about various attempts we've made, both rigorous and less rigorous, to reintroduce various herbs, particularly geophytic sort of herbs, threatened ones, but also locally rare ones, to grasslands along the Merri Creek. Just briefly, for those of you who don't know what the Merri Creek is, it's a waterway flowing through the northern suburbs of Melbourne. The catchment once contained large swathes of what is now known as grassy eucalypt woodland and natural temperate grassland of the Victorian volcanic plain. Both those ecosystems are now extremely depleted, down to a fraction of their original extents, thanks to colonisation. But we do still have some remnants of them left in the catchment. The group I work for is a environmental group formed out of a partnership of local government and community environmental groups, the Friends of Merri Creek and the Wallen Environment Group. And we've been doing a lot of work to try and conserve the things that still remain in the catchment. So the grassy ecosystems that once covered this area still exist in a few small fragments. The better quality ones are often in reserves as small as 5 to 10 hectares. There are some larger reserves as well that do have quite a number of extremely rare species in them. So reserves of around 500 hectares to 1,000 hectares. But the ecosystems themselves are much more degraded there. And even in the areas where we have higher quality remnants that are undergoing active management, whether that's through fire, through assisted regeneration, extensive weed control. While we don't have any issues with the grasses themselves persisting mostly in these areas, a lot of the herbaceous species have vanished since colonisation, whether that's through past land uses such as grazing, whether it's through introduced predators such as red-legged earth mice. There's various things that have caused them to drop out. Over the last 30 years, we've attempted to put many missing herbs back into grasslands, but we've had very limited success. And particularly when we talk about geophytes and those geophytes that don't spread by rhizomatous means, a lot of plants have very poor survival when reintroduced through classic sort of methods like planting of tube stock. And this has particularly been our experience with the daisies. One of the plants that I'll talk about shortly, Microceras scaputra, we'd plant it by the 100 and have 1 or 2 plants survive more than a year. And generally when they did persist, it was only through those planted tube stock flowering, dropping some seed, then dying. But the plants that recruited from that seed actually surviving for slightly longer periods. So gave us some clues about techniques we needed to explore. So about a decade ago, we did a lot of work based on adapting some research that had come out of Vic Uni by various researchers, including Deb, who's presenting later this afternoon. looking at small scale soil disturbance. So I guess trying to replicate to some extent the bioturbation that once happened from small mammals such as bandicoots nosing around in the soil, and potentially may have also happened through Aboriginal digging practices for some of these species. So what we were doing is finding a spot in the middle of the grassland with a small patch of weeds, knocking the weeds out with herbicide, and then churning up the soil to a bit under a foot deep, breaking the soil up to a really small crumb size, and then sowing seed on top of it and watering it in. Working together with lots of really enthusiastic volunteers from Friends of Merri Creek and local TAFEs. We monitored germinants, initially looking at a very common species of herb, the lemon beauty heads, but later playing around with other species as well. And we had really great results with lemon beauty heads in the main trial site in Reservoir. Even a decade later, we had about 80% of plots still totally dominated by the species. Within a year or two of the initial sowing and recruitment, we had mature plants dropping seed. We had some recruitment within the plots, but interestingly, we had very little recruitment going on outside the plots. So once we'd tested out these techniques on more common species, we started to play around with rarer things, including Plains yam daisy, which is a variety of Murnong, a very culturally significant species. We were fortunate to work very closely with Wurundjeri Woiwurrung elders and staff on this project to try and relearn traditional ecological knowledge about this plant. Some innovations with this particular species were the need for those strange structures on the left there, which are barriers to slug herbivory, and also about preventing trampling from kangaroos. Again, with Plains yam daisy, once we had an idea of the microhabitat it required and the seasonality of its germination, we were able to get excellent recruitment in trial plots. And again, plants persisting for 5 years, 10 years, much longer than we ever had when we were playing around with tube stock. Subsequently, we've started using these techniques on many different herbs. The seed is primarily sourced through, well, it's entirely sourced through seed production areas, some in situ ones we've set up in various grasslands, but also a lot of backyard ex situ seed production areas, which are tended to by volunteers. Our projects couldn't have been undertaken without the involvement of volunteers. We've had hundreds of people assisting us over the years, from Friends of Merri Creek, from local TAFEs, and from many other groups, whether it's tending plants in their backyard and harvesting seed from them, whether it's helping to remove the weeds that are choking out habitat for these species, whether it's monitoring remnant plants or reintroduced plants to get data on their survival. Community involvement has been an essential part of this. There's a lot, and I should point out that the techniques, although we primarily trialled them with daisies, they also work with some other herbs. The spur goodenia on the right, the Tylotus spathulatus on the left, both had good success rates, but we've had really poor success with other species, particularly lilies and peas, which probably relates to pre-treatment required scarification, heat treatment, or potentially application of fire immediately after sowing. We're now working on reintroducing threatened species that have long been extinct from the catchment. So the basalt podolepis on the bottom left is showing great results with this technique. The large fruit fireweed in the centre hasn't had as great success with this method so far, although it's still very early days. We're looking to expand to peas, looking to expand to herbaceous wetland plants. But there's also a lot of questions that we haven't really factored in at all. We haven't done any kind of testing of genetics. We've mostly used, well, we've attempted to bring together in our seed production areas the fragmented populations, the scattered populations within the catchment and occasionally adjoining catchments. We haven't done any kind of attempts to bring in climate matched stock at this stage, which is all stuff to look at in the future. Yeah, as I said, there've been so many people who've helped us work on reintroducing these threatened species to the catchment over the years. Particularly, I want to acknowledge the Wurundjeri Woi-wurrung people for graciously allowing us to share in their relearning of traditional ecological knowledge with Microceras, but the many other volunteer and community groups who've participated as well. Thank you.
I just wanted to talk to you today from the seed bank here at the Royal Botanic Gardens and acknowledge the traditional owners on the land of which I speak today, the Wurundjeri Woi-wurrung and Burrawong people of the Kulin Nation, and I pay my respects to the elders past and present and the Aboriginal elders of other communities who may be here today. I'd like to thank you for this opportunity because at the moment it's a timely symposium for us because we're bringing in population genetics and seed research teams, and we're looking at opportunities to review seed collections for translocation end use. Our seed bank is located here within the Science Division at the National Herbarium of Victoria. We're a small team, but we work alongside closely with the genetics, taxonomists, as well as people from the collection staff and education, public programs. So it really is a collaborative effort. Our seed bank at the moment currently contains about 50% of Victoria's rare and threatened flora, and the material in the seed bank is used to generate knowledge about plant requirements and as a source material, and we're using it more now for introduction programs for threatened species and really as a safeguard for species at risk. So we have a target species we collect, but we don't always know the end point of its use. So our wild collections, we aim to maximise the genetic sampling of target populations to capture that genetic diversity. And this links in very closely with our conservation genetics team. An example here is the priority bushfire species, Lobelia gelida, the snow praetor, which is an EPBC listed vulnerable perennial, and it grows in these really specialised habitats. So there's only 2 locations, it's quite restricted, and seeds limiting. So we've propagated from seed of both localities, but what we're really trying to do is build up plant numbers to establish seed orcharding and also use our genetics team to kind of look at how diverse these populations are, if at all, and capitalise on this species through, it's just a natural ability to root at the nodes. So we can build up plant populations through vegetative means, as well as trying to get some more seed collections. We use this material also for other projects, for community group reintroduction projects, if that applies, but also for other projects that we're working within the gardens, such as the Raising Rarity project. So to achieve our aim for successful long-term seed conservation, we need to collect high-quality seed, and this includes full documentation of the locality, the collected details, the permits, and collection of voucher material. So to achieve this, prior to any seed collection taking place, on-site assessments are carried out. And this is to determine the status of the target species that we're looking at, and usually under established guidelines. We map our species via the geographic data that we have with our voucher records and also the Atlas of Living Australia. So in the recent case of the bushfire affected areas, target flora were identified through this priority framework, taking into account the threatened status and the percent of the habitat that was burnt. So we wanted to determine if the species that was within an affected area actually had healthy populations outside of that area that we could also concentrate and look at seed collecting from there. Timing is crucial to successful seed collection, as is good weather and open roads. Our timing is generally based on previous observations. These are either made by our team or people in the field, colleagues, and from the data that's captured in herbarium specimens. And I'm going to keep referring to herbarium specimens because really they are the backbone of what we work on. And so before we hit the road, our permits are in place, we've notified rangers, and our seed collecting fits the Millennium Seed Bank model. So we aim to collect a minimum of 4,000 seed, of which about half of that is sent to the UK as a safeguard to spread the risk. The type of collecting bags depends really on what we're collecting. We use a very dry, breathable cloth bag normally, and that's for dry fruits and seeds. Sometimes we use paper bags for some daisies, but they can be really problematic in high winds. Just think about the Bogon High Plains. Very concerning. But we also use large Ziploc bags for freshy fruits, so petunias, for example, as long as they're not left in there too long. So then we've got mold to deal with. But so for all our successful collections, we take these plant specimens that become records of our collection and are lodged in the herbarium. And we consider these a snapshot in time because they can be reviewed for identification, for name changing and so forth, flowering time and locality, because we can't always get seed, and that's really important. So we just also need to look at other ways that we can build up our collections. And so we also look at cutting material. And an example, Neville has already touched on this with the Westringia cramnophylla, but it's an example of low or no seed set. So ultimately, when Nev and Andre were scaling the cliffs, they could only reach about 5 individuals given the steepness of the habitat. So because there was 0 seed set, they were able to take a very small amount of cutting material. And as Neville was saying, we've been propagating those, and they are now in our living collections, and we will just continue to monitor those and see how they behave. So here's some steps basically of how our seed curation process works for long-term seed banking, as well as testing for viability for other projects and also other organisations. So we return from a successful field trip. We've got our seed material that we place immediately into a dehumidifying cabinet, which is set at 15 degrees and 15 relative humidity. And we do that just to increase the shelf life of what we're working with. We need to assess all our seeds. So as we are working in the cabinet, we're coming in and out, constantly reviewing the state of the seed. Ultimately, if we're working with volunteers, we want to identify the seed, make sure it's very clear for when we're cleaning down so nothing is lost. Cleaning through a series of sieves, so cleaning through size and also cleaning through weight using an aspirator. We clean, record, count, and then we X-ray. And this is also just to look at the % of filled seed in our collection, which is then informs us if we need to go back and reclaim. It also informs us of things like predation, because that can sometimes be picked up in an X-ray, but not actually when you're cleaning. We still do cut tests where we slice through seed to have a look, but ultimately we're really using our X-ray as a diagnostic tool now just to see how filled our seeds are. We then want to know how to germinate them, because that's all part of the storage. You want to know that once you take a seed collection out of storage, you also know how to germinate it. That's incredibly important. We database, and then the storage is at -20 for these orthodox collections that we're working with. This is pretty much hot off the press. We're also recently biodiversity officers in our seed science division have started work on a spore bank for the long-term conservation of ferns and allied plants. And it's Daniel Olsen who's heading this up, and he's pictured here, where he's sorting Cyathea australis frond and basically getting the spores from that specimen, again using pressed specimens here. So Daniel's just targeting what he can collect, what would be reasonable amounts of spore that he'll be able to collect from, and then also to be able to cultivate these species afterwards. So that's really important. So he's targeting 73 species at the moment, of which 23 are very rare in Victoria. Anything that's widespread, he's going to go across populations to get that different genetic lineage. So we go back to the germination protocols. We really want to know how seed is going to behave. And this is again for our endpoint to understand what we need to do to get germination. It's not always straightforward, but sometimes it is. It's great. Things germinate readily, and that's terrific. And we can go back through our banked seed over time and see if it still germinates readily using that same protocol. Again, as I said, we use herbarium specimens to really get an understanding of specific information of where that species might be. So we can keep referring back to these vouchered material. And then from that, we also just want to know probably what kind of seed bank type it might be, because this can determine how we work with it. So whether it's a serotinous, so the seed is up in the canopy, or whether it's Geosporus, which is far more common in Australian flora, and that are released and maybe released and have a dormancy with them. So these are the information that we're trying to find out. I'll just go back there. Sorry about that. We also look at, when we're looking at dormancy, to understand how to look at breaking particular types, whether it be physical or other types of dormancy. And here in this Ramnaceae study, this is what Beck is looking at the moment. She's testing 21 species, mostly Pomaderus. And again, just to get that understanding of how seeds behave, but so also just to get that sense that are we looking at seeds that look good under the X-ray, but then in this case, they still appear to go mouldy or decay and show no form of germination. So we're really looking at how a germination test can be considered the ultimate test of seed quality, but also getting more information about how these seeds behave and how we can work with them, and looking at the type of seed and then banking those with these protocols in place. Again, these are some examples of what we've been working with to build up protocols. So just some through observation, how with the Brachyscome tagellii on your top left, this is a high alpine species. We've been looking at its germination response over quite a large temperature range, and then comparing it also to seed orchard collections. We're finding that those that we have been actually growing down at Cranbourne, which is at 90 metres above sea level, is really germinating well. And so we're really quite happy about how that's progressing, building up our seed collection of this particular species. We've looked at scarification for very rare species, and also hot water treatments, as in the Pomaderus vicinifolia. And we're interested in this because we're looking also at how Beck's work will correspond, where she's using a dry heat and temperature. So when selecting these germination conditions, we really want to think about these natural environmental cues and how that might stimulate germination, and the conditions and the dormancy breaking treatments that can be used to overcome these seed dormancy. We also use climatic data to determine optimum germination temperatures. So as you can imagine, this germination work is ongoing. We're always building, trying to work out different types of dormancy alleviating work just to try and stimulate germination. It is incredibly ongoing. But when we do have germination, we basically try and keep all those seedlings and grow them up. And so from our germination testing, we get these living collections that we can then add to our state botanical collections or through our living collections, or we pass on to interesting groups. And for example, anything that we do at large scale, like in Beck's Rhamnaceae study, we will then offer those seedlings out. An example here we did with Callistemon Kenmorrisii and Forestiae. These two species had, after the recent bushfires, were critically endangered. We were worried that they could become extinct. We could not collect seed at the time. It wasn't feasible. We couldn't get out there. So we actually went back into the seed bank and took our older collections out and looked at their viability. We tested them with a couple of other closely related species, and just to see how they behave, were they still viable, and then to really get an understanding of what would be a really good temperature for best germination. And from this work, it really complemented our bushfire recovery efforts. And we were able to, from this work, generate a lot of seedlings, which we were then able to provide to land owners, to the Far East Victorian Landcare Group and Muji Aboriginal Council. So then taking these plants and putting them back out there to further support the bushfire affected areas. We're also concentrating our efforts on germination responses to inform rehabilitation programs. And this one in particular is for direct seeding and practical rehabilitation in the Victorian Alps. So we're looking at how particular endemic species are going to behave and looking at their germination responses. And again, with this information, sharing it through Hotham and the Falls Creek groups to really get these metrics out to determine a lot more about how these target species, their early life strategies, and how they may respond. So this is a very exciting time. Over the summer of and autumn of 2022, this year, we've collected 164 collections across the Bogon High Plains of 34 taxa. And with that, this will all become informative to looking at how we can do direct seeding in rehabilitation efforts up there. We also work with organisations such as Glenelg-Hopkins CMA, and here we've been assessing the button wrinkle world. So this is from their in situ collections. So we've been given seed that we then test the viability and then germination. And we've really been able to provide this information back to Glenelg-Hopkins on how those populations are behaving, and the seed is really viable. So it's really terrific for us. It gives us additional information too, because we grow things on, and then we're able to present those in our living collections and look at how they perform over time. We've also explored how they perform in potted environment, as well as in the gardens throughout our different living collections at Cranbourne as well as Melbourne. And with our recent funding and partnership with the Glenelg-Hopkins, it brings us to, again, looking at working with a particular species where we can look at seed collecting and surveying and then getting an understanding of this species. And this is from specialised saline habitats. So we can really look at the threats that the altered hydrology and weed invasion, and through this partnership, look at how we can build up populations, do some genetic work on the diversity across the 11 important populations within Glenelg-Hopkins, as well as identify new sites through habitat and site assessments. So it's an exciting time for us where we're starting to collaborate more, doing a lot more with our germination work and trying to really start getting some partnerships and opportunities to work with people to get plants back out and using our from the information that we generate here in the seed bank. So thank you for the opportunity to talk today.
Thank you, everyone who presented, Sarah Jane, Richard, Lucy, Jen, Neville, Michael, and of course you, Meg.
And learn about the propagation methods. So permits, as have been mentioned, are necessary for our rare and threatened taxa and need to take that into consideration. Do you actually have to get permits from a state level, federal level, or even just from private landholders to go onto the land? Accurate records, again, reinforcing what Meg said. Herbarium specimens are imperative of collecting rare and threatened material just to validate that you've actually got the correct species that you've been propagating. Strong records of where you've been collecting the material, so provenance information, any treatments that you do to the plant material from time of collection right through the propagation methodologies to planting out, and then ongoing management if you're doing in situ revegetation work, ongoing management records so that you know what is happening, the sorts of messages and learnings that have been achieved from Merri Creek. It's worthwhile making sure you've got good skill and resources available for the propagation, but you know, things are not completely requiring a high knowledge level. You can get away with relatively low knowledge levels, but just have basic information available. And make sure you program your growing of plant material so that you're actually having plant material in prime condition ready for when you're planting it in the ground. Make sure your site preparation is going to be up to date and getting rid of weeds, etc. Do you need vermin-proof fencing, etc.? And time your planting. Obviously, don't plant in the middle of summer, things like that. Some of the tools you might need for propagation or collecting cutting material and/or seed in the field, snips, various types, other propagation tools, including scalpels, very sharp knives, a sharpening tool to make sure that your knives or scalpels are always kept sharp. Clean cuts give you a much better healing capacity on the wounds, and there's less chance of fungal infection. So hygiene in a nursery situation, good hygiene is a good indication of a reliable nursery grower who can actually provide the resources that you need to grow plants on successfully, and labeling, recording information about your stock all the time. You don't have to have a high quality mass production nursery to grow plant material. You can have simple little timber structures with plastic cover going through to then below the glass houses which have got a quality production standards and custom built, or into the booths where you can actually have plant material being grown in specialised climate control booths, which give you much better potential survival rate in some very difficult to grow taxa. Through from the top of the levels and fancy materials, you can go through to very basic growing tubs with plastic bags over them, styrene boxes with the top cut out and a bit of plastic film put across the top, through to commercially available growing containers like the two illustrated at the bottom of the page, which you can go down to your local good garden supply or hardware store and usually pick these up. One thing about the plant propagation box in the top left corner is that they've used sticks to hold the plastic bag in place. Those sticks could be carrying fungus or other pests, and that might be introducing the pests or disease into your cutting material. So don't use sticks, just use be it plastic or bent wire hoops, something like that. Back to seed, as Meg was referring to earlier on, collecting of good seed is important. So the Xerochrysum pilastri, which she had slides of earlier on, these photos were taken by Sana, and the population on the left, one population of the Xerochrysum on the left had excellent seed germination and capacity, and good viable seed, whereas another population, which was a fairly small population, isolated, had practically no viable seed in it. So when out in the field actually doing the collecting, make a point of looking at the seed in the field at the outset, just to make sure that you're not just collecting seed heads, which are not going to be of any value. In the bottom right corner, I've got the GG with the chapter 7. A lot of these details about this sort of information in this part of the symposium are referring to various chapters within the germplasm guidelines and also the Translocation Guidelines. And they are two worthy documents to use as reference for any collecting you might want to be doing. Once you've collected the seed, germination in bringing them back to the nursery, you can grow the seeds on. Aim for a non-soil based substrate, and ideally with practically no organic material in it. One of the biggest problems in glasshouse and growing conditions is the fungus gnats. Fungus gnats will go into your potting mix, and as much as they do break down the organic material in the potting mix, they also do attack the young roots of seedlings. So control of fungus gnats, so coming back to hygiene, is very important. The other part of it is actually selecting and pricking out your seedlings at a very young stage. Usually seedlings are pricked out at their first pair of true leaves or second pair of true leaves stage, and usually handling the young seedlings with their cotyledon leaves, because they will die and eventually, or quite quickly anyway, and it doesn't matter so much if they are slightly bruised in the handling process. Sometimes you'll see in less suited nurseries for your production, plants that these have been deliberately left in the tray, but plants that may not have been pricked out early enough, seedlings, they might look fine and healthy on top, but when you look at the bottom of the tray of seedlings, the knot of massed roots is making it impossible to actually successfully prick out these seedlings, and the damage to roots that will occur, you will not end up with a good root system on your plants in the future. So this sort of material should be just thrown out. Don't even hold it typically on the off chance that it may be used in the future. On to vegetative cutting or vegetative propagation and cuttings at the start. A range of different sorts of cuttings you can do. So hardwood cuttings, softwood cuttings, and there's medium or semi-ripe wood cuttings. And the type of cuttings you would actually take will vary depending on the plant species. Some plants will strike very readily from cuttings, others are very difficult to grow. The cuttings here is a semi-ripe wood cuttings, and you can see that where the first on the left, the cuttings have been cut off just below a node, and then the lower leaves have been trimmed. This is a heel cutting where you've got a main shoot and side shoots are pulled from the stem of this main shoot. They get pulled off with a small heel in the middle photo lower down. They strip off a bit of bark from the main stem, and then that gets trimmed. That heel is trimmed up to a bit of clean wound, using a sharp knife or scalpel. So the ripped off bark is completely trimmed off, and then leave, in this particular Syzygium species, the leaves are trimmed as well. The advantage of taking off heel cuttings or the cuttings, tip cuttings at the nodes is that there is still unspecialized cells present at the base of the heel or at the nodes from the cuttings. And these unspecialized cells are what will form into your root primordia and develop the root in the root system. So in the blue on the left, you can see the roots actually breaking out through the bottom of the cutting and starting to develop into quite a good root cutting. Root primordia start off with just a single cell and then multi-cell proliferation. And the primordia usually don't have the strength to actually push through the bark. So people often think about the bark, that the roots come from through the bark of the stem, but they don't have the strength. They will actually come through the base of the cutting. So sometimes there is value in some species to actually trim a small amount of bark off the sides of the cutting as well, which makes it easier for the root primordia to break free. Often in the lower left corner, you'll get callus developing, and the callus material will help keep the cutting in pristine condition and help absorb a bit of moisture, but it actually inhibits the root primordia breaking through. So it may be that the root, you won't actually get the cutting to strike roots. So it's necessary to trim the callus off from the plant material. People ask often about what size of cuttings do I do. One of the big concerns is what size of plant are you taking it from? The grevillea in the main cutting, you can see the cutting material is actually about 200 mil long, and the size of internode influences what you could actually successfully grow. Whereas up above the Uartia and the Nephalium in the little small image at the top, the internodes on those is probably a millimetre or less. So you could be typically taking a cutting that's only 10 millimetres long. So there is no given set size for taking cutting material. If out in the field and you're going to be away for quite a few days, it's often valuable to actually treat the cuttings and prepare them in the field rather than just storing them in plastic bags. RBG has been recently involved with collecting in far north Queensland, and we took with us some materials which we'll talk about in a second. And our survival rates of cutting material were in excess, far in excess of cutting materials taken by other botanic gardens which are on this multi-institutional project. One species in particular, we had survival rate, whereas no other organisation has been able to grow the particular plant, which is a very rare Syzygium species. So preparing in the field, we've found it highly valuable to take with us the preformer plugs, which are a mix of peat and coir. And these act like a small vase and hold moisture more strongly around the base of the cuttings. And you can prepare your cuttings, put them straight into these preformer plugs, and be able to, the plants will actually start producing the root primordia, the cuttings will start producing the root primordia at a very early stage, and it'll potentially increase your survival rate. In this particular main photo, it's demonstrating the values also of using growth stimulants to enhance the root development. The value of these plugs is that you can actually prepare them in the field. They can be stored in plastic bags, which makes them easy to transport. And when you get back to the nursery, the plugs can be going straight into a glass house, and the cuttings themselves are not disturbed. So you've already potentially gained several days of potential cutting establishment. Other forms of vegetative propagation, if you're out in the field and collecting from a location where you know that you'll be returning and it's plants that may not grow successfully for cuttings, it is possible to do field layering. So using small side branches, put a small nick in the bark where the red circle is, then use a couple of wire pins to pin the plant down or the branch down, and then pull the soil back over it. Ideally, the soil needs to be a reasonably open, friable topsoil. So sometimes in very rocky situations, it can be difficult to find a place to do that. But one of the advantages, particularly with Ericaceae, that you can actually find a lot of Ericaceae like Acrotriche and the Astrolomas will actually produce aerial layering in the soil anyway. So you can actually trim off side branches and transplant side branches back to the nursery situation where they can actually be propagated up more successfully in a managed environment rather than out in the field. Back to the xerochrysum we had the photos of earlier on, and the what Meg was talking about too from the seed collection perspective. Where it's difficult to or impossible to grow the plants from seed, they can also be grown from division. So division is generally referring to stolons which have got a root system already established, but rhizome cuttings are cuttings that or sections of rhizome that don't have roots established on them. So out of this one side branch on the left, it's possible to get a number of different rhizome cuttings, ones with the new shoot where you can see that there's a small amount of root at the base of the cutting, and that small amount of root is indicating that the wood is hardening up and happy to produce roots. So that shoot could be put in as a cutting. The older brown stem section below it is an older shoot which will have dormant buds in it. So it is still worthwhile putting in as a cutting and trying to grow plant material from it. In the main image that you'll actually see a section of stolon where the material has started to blacken off and die. That material may do something, but it's pretty unlikely to establish. So it's worthwhile putting it back into the soil where you've had to lift out the cutting material from out in the field. Rhizome development. So probably the size of rhizome here, illustrated here, is probably a lot larger than you would be growing typically in Victoria, but certainly relevant for collecting New South Wales and further north, but also relevant for growing things like lomandras, etc. The same principles apply. Dig out a section of the side of the plant and you can quite successfully divide up one division of, or one cutting of, or one section of plant that you've dug out into a number of different units which can be propagated up and will grow successfully. Certainly collecting in the field can take a bit of experience and knowledge. And this is where I say earlier on about learning from those who have dealt with plants before. That, for instance, the Linus spadix here, a rare palm from the high country or the high mountain regions of far north Queensland, very rarely sets flower and sets seed and has not been successfully propagated before. This particular plant, I was looking at it with the team and they were all pondering how to actually propagate up cuttings and so on. And I just said, well, have you got an axe in the back of the vehicle? So brought the axe out and took a rhizome division from the plant. And those rhizome divisions have grown successfully through to the collecting of seedlings at the other end of the other extreme, using a penknife to dig out minute seedlings of another endangered species from that far north Queensland material. Vegetative propagation using grafting is a lot more of a specialised task, and it's something that should be done in the nursery. A range of different grafting methods are available, and certainly a lot more research can be done on that, but this is where you need to be looking and getting advice from specialists. The graft on the left, the cutting graft, is a cutting that has been taken and put into the preformer peat plugs that I was talking about earlier on, and then the graft has been put straight onto the top of the cutting so that the two of them can get established at the same time. Seedling grafts, where the seedlings are actually grown on and the graft is put in the nursery. And then once the grafts are growing successfully, you could end up with the sort of graft illustrated on the right. Grafting has been used a lot in WA for grafting of eucalypts and corymbias, and illustration here on the left of Physofolia, the red flowering gum on spotted gum, and on the right, a very good form of spotted gum onto a seedling of spotted gum. And they seem to be growing very, very well. The one on the right, the spotted gum, was in a group of 3. 2 of the plants were okay. The third one was like this on the ground following a windstorm. And if you look closely at the top right corner, the red circle indicating some sawtooth-like protrusions from the base of the stem that's broken off, that's the only tissue in that graft that has successfully bonded with the seedling stock, the stump that's still left in the ground. You can see that the rest of the tissue is really quite smooth, and there's been no proper bonding of any of the woody tissue between the two. That basically there's just been a cambial transfer of liquid and nutrient to keep the plant alive, and that's it. Once you get to the stage of planting out, you should have had all your plants growing on successfully. Your site preparation should have been completed, and things like the fencing, getting rid of weeds, so on, understanding what vermin are going to be around. Is it the right location to do your planting, as Neville was talking about with the orchids? But one of the things to look at is make sure your plants are actually in good condition for planting. The one on the right, the roots are starting to have browned off and have hardened and become quite established in the pot. So it'll be very difficult for this, a lot more difficult for this plant to establish a good root system that will grow well in an in situ location. It might be right ex situ, where management can be much higher, management levels, but if you're looking at revegetation and restoration, then that plant might be problematic. Ideally aim for plants like the one on the right, where the roots are quite white coloured and growing very vigorously. They will naturalise into the soil quite well and spread, and they will go down deep, so you'll end up with a good, well-established plant. Some of the stock, this again, plant has been deliberately left as a plant so that it could get pot bound. A eucalyptus sapling. This species had swellings around the base of the stem, and many eucalypts, especially non-mallee types, this is an indication that the plant has been pot bound and not healthy. The other part of it is when the plant was, as a seedling, was pricked out. It was put into a hole in the potting mix in the small tube, and the roots have been twisted up. So you can see there a J root has formed. The J root is problematic. That is potentially, you'll end up with as the plant matures, that the plant will not give you the good growth and established root system that you will want. And you could end up with this sort of situation. This tree, you can see the scale of the radio, was over 200 mil diameter near the base, and the roots had girdled around from the very early potting stage. The roots had gone around and around and just strangled the plant. And if you look at the base of the plant, the black area at the base, you can still see the shape of the pot where the plant was knocked out of the pot and planted in the ground. So not the sort of thing that you actually would be wanting to be growing in a revegetation habitat. And micropropagation, so what has been alluded to already, and more talks later on about micropropagation that's going to be occurring and can occur. Again, very specialised laboratory works, and it would be something that you would need to coordinate with other growers. So, thank you, and I've put the web addresses on for the germplasm guidelines and for the translocation guidelines, and hopefully you'll be able to download them if you haven't got that information already.
Hey, today I'm going to give you an overview about Pimelea spinescens, which I spent the last 14 years of my life looking at. Summary of the translocations and what we've learnt, and ending with the recovery team's recommendations and what we think are best methods to establish transplants. Give you a bit of an overview of Pimelea. There are 150 genus Pimelea worldwide. It's found in New Zealand. There's about 36 are endemic to New Zealand, 110 are endemic to Australia. So it's a Gondwanan species, and about 18 of these species are considered rare or threatened. Pimelea spinescens subspecies, its definitive distinction is its spinescent stems. They're not spines, they're just pointy stems. And there's two subspecies. We've got spinescens, both are endemic to Victoria, and there's also pubiflora, which is distinctive feature is its hairy seed pods, as compared to spinescens having quite glabrous seed pods. Also, pubiflora is a bit larger plant, about 50 centimetres high, and has quite yellow flowers compared to spinescens being creamy to white sort of flowers. The distribution of spinescens should be in all those beige areas that you can see there, retracted back to green, and we know it to be in the red areas, which was the 1990, this is a 1990 picture from DSE, DELP, where we think it should be. And it's both it and its ecosystem are critically endangered on the federal EBPC Act, and federally it's, statewide threatened. There's a recovery plan of which we're currently in the process of revising, an action statement and conservation advice. Also, pubiflora, just wanted to show you where it is in the scheme of things. It is only found through two populations that we know of. It was found to be extinct. We thought it was extinct in 1900s. We hadn't found it since 1901, but in 2005, there was some roadworks on Lake Road, Natimuck, and about 350 species were found. They're still there. And also Minyip is another area that we found it, with 191 plants found a few years later. Here is a bit of closer look at that area. And then with a bit further looking and a bit more investigation, there was another small population that's been found in Kalkilly East, very tiny. So it's in a very small select area. So very, very rare and threatened, this one. Okay, spinescens and most Pimeleas are considered dioecious. So that means that each individual plant has a separate sex. To the right of the slide is the male, with the obvious stamens and the pollen on view there. There was very showy plants in the landscape. And to the left, we had the female with her anther, and she has no pollen on her stamens, although she does have stamens. It's quite a small shrub, only 5 to 30 centimetres high, can be prostrate or upright. There's another picture of spinescent pointy stems as such. This is a classic presentation in the wild or in its natural habitat. To the right, we have the female. She is in full flower there, and she hangs all her little flowers down. So she's sort of saying, don't come and get me. Whereas the male over on the right there is saying, here I am, come and get my pollen, come and get me. And we think that that's due to probably its spinescent stems in it that's highly palatable. It's very nutritious, this plant, and things would have been eating it. So the female's trying to say, don't eat my seed, and the male's saying, it's okay, it's all right if you eat a little bit of me. It's probably what's going on. It produces seed during winter. So it flowers during winter. We can see it flowering in Melbourne from about February, but towards the Western Plains, it probably flowers from about April through to August. Insect pollinated. We think it's mainly flies. There's about 1,000 different species of fly, which we would all attest to. And it's quite generalistic, essentially. Insect pollen. It produces a droopy fruit, which is like a peach or a pear. And this is what I have seen. It's very rare to see this. I've only seen it a few times, but basically that is a white fleshy fruit outside. Normally it's quite a dry looking seed. It is oily. Pimelea means soft and lard and fat in Greek, nutritious, and it will float essentially because it is an oily seed. We're very lucky with Pimelea that we have a very active recovery team. Since about 2007, we've been having meetings. There's about 2 or 3 a year with many, many stakeholders coming along. They get an opportunity to get together, inform each other, discuss, we put together protocols such as the Translocation protocol, and we've put out best practice. So we share technical advice, and we have members from all different areas, DELP, consultants, SWIFT, Parks Victoria, and there's 24 councils across Victoria that have Pimelea spinescens in it. And we're also very lucky to have some funding, which unfortunately came from mitigation. So it was established following an infringement, and we had some funds put in Trust for Nature, who administers it, where we give out funds annually to conservation reserves, and projects can apply for this annually. We've funded to date since 2011. We've supported a PhD that I undertook many years ago now, investigations into genetics, investigations into translocations, many private landowners, land care groups. We've also looked at direct seeding, and also given it to Ben for the common to look after managing that really large site. So a lot of different research projects have been funded. Liz James Genetics basically looked at whether we could mix plants, whether it was a good idea to mix seed plants and do seed orchards. So in 2012, she looked at the genetics, and from many samples across 20 populations, she found that essentially pubiflora and spinescens are distinctly different. They cannot be mixed. The populations until quite recently were actually very well connected. So plants within 50 to 60 kilometres were more genetically alike than we would have expected. So we wondered why. Was it plant longevity? Is it because there's a really great seed bank going on? And could it be moved by birds and also floods because of its oily seed? So what she did find was all the plants from essentially Melbourne and the plateau around Keela, across the Werribee Plains through to the Western Plains can be mixed to a degree, but the plants beyond the Great Dividing Range have been separated by quite a while, and you wouldn't want to be mixing those plants in. They're found on the black clay soils up there. So through my PhD, I was lucky enough to give an opportunity to dig up plants, well, maybe not lucky, but these plants would have been destroyed anyway. Not all plants are going to be saved at this site. So I dug them up, and we found that they had a taproot. We didn't know this essentially. People thought they could have been rhizomatous. They weren't quite sure what was going on. When did a plant stop and another plant begin? But all of them had this enormous distinctive taproot, of which you can see when you do a translocation. If we get the bottom of that taproot, we would get most of the plant, and they do actually make it. The root goes down about an average of 50 to 60 centimetres. This is also how it's in the soil structure. It sinks over time, that taproot. So the taproot's quite low in the soil there, and we think that's because it's living in clay. In summer it's like concrete, and in winter it's like it's quite soft, so the taproot will sink lower in the soil profile, and that's why sometimes when soils are scraped, that plant can still be surviving later. Here's another picture of what you might have thought was actually not part of the other plant, but it is. It's all part of one plant. It's just that these little underground stems can grow at all different levels. This is a plant that we dug up. It was the largest plant in the area. And when we got it out, we found that the lateral stems extend, so lateral roots extended up to 1.8 metres one way and about 1.5 another. So it's got an extensive root system, but still that taproot system prevails. This is that plant dug up, and there was a rock in that spot, if you're wondering why that different root came out at a different level, but there's a lot of roots going on, and they don't compete with each other, this plant. The next slide will show you that we thought this was one plant, but if you have a close look, it was actually 3. Go, and they're all quite happy, safe little plants, and they grow this distinctive taproot to start off with to get down to that clay soil. Major issues are that's very fragmented in large numbers and size of populations. Recruitment has barely been reported. We didn't know how long plants lived. What is their natural recruitment weight? Only 5% are protected in reserves, and the current threats of roadworks, cemeteries, industrial, agricultural and urban development is ongoing, and definitely lack of management in a grassland, lack of fire reduction. So we also were able to get a translocation review done in 2013 by Biosis, and that looked at the 11 translocations that had occurred. And we found that generally, we're going to move, why isn't it moving? Oh, there we go. The aim of the review was to summarise the methods and survivorship and outcomes of these translocations across all these sites, provide an update and compare survivorship of this population. Basically, we found that the average survivorship was about 33% across these sites. General trend was decreasing survivorship with increasing time, and the initial loss of plants was quite large or could be small depending on how it had gone. And there was an overall trend in gradual decline in survivorship that followed, unfortunately. The causes were essentially a lot of it to do with lack of biomass management, weed encroachment, high biomass at many sites, inappropriate fires, rubbish, inappropriate fire management, and there was a lack of monitoring. We realised people weren't reporting back, and a lot of this information was only a couple of years old, essentially. This is one of the sites that it actually got translocated. That's Christie's Road near the Western Highway. This is the conservation reserve that was left. All the green where little crosses are Pimeleas, and the red ones were the ones that were actually translocated into that site. Also the hatching, just to note those hatching at the side there, that's where the translocated plants were planned to be translocated into. There's a few photos of translocations going on, which Steve will talk about a bit later. That's the site after it had been done after a while, and you can see there's a vague fence in the middle. That's that line in the middle over there where all those plants were. Unfortunately, the site was still going to be, buildings were to be built on that site, and it hasn't been actually maintained for the buildings. So when the buildings were going to go in, they levelled the site, pushing all the dirt into the conservation reserve because the fence was almost non-existent. And unfortunately, a lot of those translocation plants were sitting right there, plus a lot of the natural ones. Also, you get, unfortunately, shading due to the buildings being built right up to that fence line to maximise their space, which is now a new problem in that grassland. That was post the remedial works being done, moving all that soil out of the reserve, and some of the plants did live after that. Translocation of all plants is worthwhile, essentially, because 40 to 60% survive, depending on management. If the plant dies, the plug still holds a seed bank, and there's probably a mycorrhizal association going on, which is great. So we then looked at cuttings. Cuttings, unfortunately, 90% of the cuttings haven't worked essentially over time. They don't grow the right taproot, as you can see here. We looked at seed. We tried to do some direct seeding, but to date, that hasn't been very successful, unfortunately. And we wanted to know, did they survive? So in my PhD, I was lucky enough to get to look at how to germinate these species. So I looked at about 27 different treatments and found that essentially anything with gibberellic acid would promote germination, which is basically a plant growth hormone that overcomes the inhibiting hormones that are in the seed, because this seed has a seed dormancy. And you can see that quite clearly with the timing. I looked at a few different incubators, and we started an incubator in summer and an incubator in winter. And we found immediately heading into cool conditions, we've got really great germination spikes, which tapered off over during spring and then stopped altogether in summer. But when I left that particular lot of dishes in the incubator, and all I did was add water over time, there was nothing else done, we got another germination spike the following winter when we went back into cold conditions. So even though we had not all the viable seed that was actually viable had germinated in that first period where we had perfect conditions for it. So the seed is hedging its bets and germinating when it feels like it. So you won't get all the germination happening immediately. So collecting seeds worthwhile. Seed bags go on about July and August when you've got seed on the plant. The seed will have plant will have seed very early on. August is the best time where a lot of the flowering's gone and there's plenty of seed. Pop your little bags on and collect them in about November, October. Once it's warm, when it gets warm, the seed will automatically drop off this plant. Otherwise, it's quite green and it's still probably photosynthesizing on this plant. I use an ice cream container. The seed should be stored over the summer period because it'll never germinate then. And it's great if you expose it a little bit of warmth, because the seed would normally have that happen if it's lying on the ground over summer, very hot conditions in a grassland. It can germinate up to 6 years after post-collection. So the following March, I'll get my germination out, 3 months of plantings, treat with gibberellic acid in little Petri dishes, and then into incubator with cool temperatures, so average 5 degrees at night, 13 during the day, and just add water as required. Here's a few dishes. This is us setting it up. That was my incubator that we had at the university, and we packed all the little seeds in their little beds, put them to sleep, and then if we're really lucky, we've got lots and lots of germination of the species. So then we potted them up, put them in their little in the little trays. You can see the seed, the root bolts. It wants to get its little root down as quickly as it can into that lovely soft soil during winter and get maximum growth before summer hits. More pictures. So I want to figure out, when you do plant this plant, what's the best way to do it? Should I plant it a grassland with a Hamilton tree planter, or should I put it into a really nice ploughed area with lots of forbs? Because forbs, grassland forbs, form a taproot as well and are not competing with the plant. We also then tried treatments of burn, mow and control just to see if that was going to make any difference. So here's some pictures of the plants we put in the grasslands in September 2013. This was the first chance I had to do it. We had quite good success for 1 year. We had 2 plants surviving of those 9 plants. They did really well, and they're still surviving today. So we can do it. This is a really amazing thing to see. We weren't sure, and there was no plants actually at this grassland, so I was quite worried that it wasn't a site with plants already in it. And then we tried the ploughed and sown into September 2012. This is a site that had been cropped regularly. Greening Australia dug it over the soil. We've popped into forb seeds, which took a few years to come up, so we didn't want to plant into it just yet. But over time, we got an amazing coverage of grassland forbs, and it was also weeded a lot, so it was kept clearly open, all natural without grasses. And then in 2014, I was able to plant 5 plants in the plough and 15 plants into the grassland. And over time, that's the grassland to the left, which is 1 plant survived, unfortunately, but half of the plants in the plough plot survived. So they did really well. And this is the results after a few years of doing this, that we found that 22% would survive in a grassland, but 41% survived in the ploughed wildflower. So it was a much better success rate in ploughed areas. So if you can dig over that soil, it will do well. There's some pictures of the September 2015, the plants there, and then it's exactly the same plot down the bottom in May 2018. Those plants are doing really well, producing lots of flowers and lots of seed. And what is the real icing on the cake, which I've heard a little bit from other talkers that you don't necessarily get, is we got germinants. So we had at least 1 germinant that has germinated, and we're hoping for many more, of course. And that was in the plough plot, which was kept really open, and it's still alive today. So this ecosystem, this area is now producing its own germinants, and hopefully will continue to do so. So there's 160, I think there's 41, 160 down the bottom there that survived. So since this research, plantings have now occurred at many sites. We've done it in translocations because we have in the translocation protocol, we have a stipulation that if you lose more than 50% of the translocations, you need to grow plants and add them. So Pioneer Park and Carolyn Springs Railway Station have had additions. That one's at Pioneer Park, and was done in 2019, and that picture was taken the other week in 2022. So it's growing and adding to that population. We also did it at Skipton Common, adding and augmenting new genetics to that area with a whole lot of school kids who are out there looking after it. And recently, we also did pubiflora. We grew a lot of pubiflora seeds, or a lot of pubiflora plants, which did really well. And we, in the middle of the pandemic, got them up to Minyip, and they were planted by locals into the Minyip site. And today there are 55 alive. So we can augment small populations, and we can move things around, ensuring that genetic mixing to future-proof sites and saving the genetics at risk. A lot of plants are at the sides of the road, and they might not be there much longer. So we can collect some of the seeds and grow them on and put them into secure sites. I think that's the future for Pimelea spinescens. Okay, thank you. Is there any questions?
Okay, thanks for that, Debbie. That was great. Now I've got to work out my technology. So the practicalities for the salvage and re-establishment. It's not a bad title there for the re-establishment of one of Australia's most translocated plants, but to some extent that's unfortunate. But yes, it's to some extent inevitable now. How do I do this? Space bar. No, it's not that. It's not that. Just click on left click, is it? No. there's our acknowledgement to country. Thank you very much to the traditional custodians of the land. And moving on. So why would we indulge in translocation as a mitigation exercise? Particularly around Melbourne, development will continue to occur. All of our individual plants are considered valuable. We're a critically endangered species. And in that context, well, there are a defined number of populations that we're aware of, and very few of them are actually increasing in size. They also then offer the opportunity that over and above the loss, if we translocate into a secure site, that there'll be actual resources for management for that site. And we also get to learn about the species biology. So as I've indicated, we live in a highly regulated environment, and development is a constant feature of our urban environment, and impacts to biodiversity are going to happen. We might not like this decision, but as you can see by the gentleman in that photo, you can complain about decisions until you're blue in the face. You don't often change things. So impacts to native vegetation can generally result in an offset requirement, but these typically don't impose any requirements in relation to the species that are impacted. And so an exception to that is within the Melbourne Strategic Assessment Area, where there is a requirement to salvage, but to date, DELP as a regulatory authority hasn't really salvaged any of the Pimeleas, and they have tended to be bulldozed rather than anything else. So translocation offers us an opportunity to mitigate approved impacts to threatened species. So again, going through that again, Pimelea, it's critically endangered. Recruitment is rarely observed. Propagation is difficult, although Debbie's been getting on top of that. And nursery plants have been difficult to establish in the wild in the past. We're doing that with the recovery team a little bit more successfully now, but still at the end of the day, it's still a fairly tedious process. And so the western fringe of Melbourne, which supports important populations, is still subject to development pressure, and we still get regular approvals to destroy populations of this species. So a mitigation translocation, as we've heard, attempts to establish plants that would otherwise be lost into new locations so that we can retain that population. As Debbie's pointed out, plant cuttings have been pretty unsuccessful. Collection and propagation of seed can be done, but time's often limiting. And the physical relocation of plants has been relatively successful. It has its limitations, but also it has its benefits, that once you've moved a plant, as long as it lives, you can still actually collect seed from it. So the issues for translocation of Pimelea is basically the form of the plant. That fairly sturdy taproot goes down to a depth of about ½ a metre, as Deb's pointed out. The physical environment that it grows in, it's a fairly heavy clay soil, which is basically concrete during summer and relatively soft during winter when it's wet. So when you want to move a plant, you have to be very careful of the level of disturbance that you're going to create in the place where you're going to put it. So if you're going to put it into an area of relatively intact grassland, you don't want to be driving around in heavy machinery because you'll end up destroying the place. Having said that, there's a relatively limited availability of recipient sites. But while we do have a number of small grassland reserves that things can be translocated into, management of those sites is something that translocation provides an opportunity for. So even if a translocation ends up failing, you end up securing significant resources for the management to try to keep any such site as a good grassland. But those types of recipient sites do need to have been secured. The timeframes for management and monitoring that we've come up with is basically a minimum of 5 years. Earlier on in the process, for many translocations, that was only about 2 years, but we've found that that's just not adequate. You can't work out whether plants are recruiting in that time frame, and look, sometimes even in 5 years you can't find that. The issues that you need to resolve before you do a translocation is that you need to prepare both the salvage site and the recipient site. Counting how many plants there are is often awkward. Grasslands, particularly in development sites, are often pretty dense, and the plants are quite small, so being able to see them can be quite difficult. So one other thing that you need to consider in your recipient site is how many plants are you going to be moving, because you need to have enough room to put them into. And again, you need to have enough disturbed area to try to put them into, because we have focused on putting plants in disturbed areas rather than into the middle of good quality grasslands, because it's just too difficult to get into right into a grassland without causing any damage. So we try to do some level of weed control at the salvage site and at the recipient site. We do it at the salvage site largely around the plants before we pull them out of the ground, to try to minimise the amount of weed material that we move with the plants, and initiate weed control at the recipient site, so you get a bit of a head start on controlling the weeds once you put plants back in the ground. As far as plant diseases is concerned, similarly to weeds, we don't want to be moving problems from one place to another. Fortunately, with Pimelea, we don't seem to have a lot of soil-borne disease issues. The next thing we have to consider is how far we're going to be moving plants. Early translocations were relatively simple. There was a fair amount of in situ protection for parts of the population. Normally, larger concentrations of plants were protected, and other plants that were more remote from those concentrations were basically within an area that was approved for development. And it wasn't much more than 100 metres or so for plants to be moved from the development footprint into an area that was to be protected. Subsequent to that, one of the examples that Debbie put up was for the Deer Park bypass. And in that situation, plants had to be moved well over 1 kilometre. So when you see the methodology that we've used, it's quite awkward moving one plant at a time if you've got to move it a number of kilometres. And so you had to work out a way on how to hold a plant and transport it to the new location and then put it back into the ground. So all of those things come to evaluating the amount of time that you need to do the work that's needed to move the plants. And you also have to make contingencies for stuff-ups. And in that process, you need to have a very clear plan. You need to have a very clear idea of exactly what you're going to do and how you're going to do it. Because if you don't know that, it's going to take much, much longer. And these as mitigation translocations that are basically funded by a developer, they're normally pretty keen for you to get in and get out as soon as you possibly can. And obviously want to minimise their costs. Again, yeah, if you sort out exactly how you're going to do things and how to do things properly, then you can cost things appropriately. Now, for the physical translocation of Pimeleas, we generally do these during dry conditions, and we do that again because of the machines that we're using are pretty heavy machines. And we want to minimise the impacts on the areas that we're actually taking plants to. So fortunately, the basalt soils absorb water very efficiently and very quickly. So we can pour water on a highly localised area, it gets absorbed into the soil very quickly. And then the soil becomes soft and plants become pretty easy to remove with the machinery that we're using. So the machinery that we use is called a tree spade. It's mounted on a tractor. And when you're not moving plants very far, you can actually replant into a hole cut previous to the salvage cut. You can plant directly back into that hole and minimise the amount of handling that you've got for that plant. If you're moving it at a considerable distance where you can't just drive the tractor back and forwards, we've been placing plants into a supporting basket, and that's a steel mesh basket, and it's basically lined with hessian, and the plants placed into that and then supported during a mass transport exercise where you pick up 20 or 30 plants at a time in a trailer and then transport them over to the recipient site. So plants are then put into a hole cut by the same type of spade, and you fill up any gaps and water liberally, because basically the plant, it being translocated during what was otherwise dry conditions, has suddenly been watered, and it suddenly thinks there's lots of water in the environment, and wakes up from its dormant slumber from the summer and starts growing. And if it becomes dry again, then that shock will most likely end up killing it. So here's a movie that a number of you people have seen already, but some of you may not. So this is how our tree spade works. Oops. And you could see here at the end of that particular film, he was opening up the spade to allow the plug of soil to be exposed at the base a little bit. And what we normally do is we trim the absolute point of that so that it sits in the hole where it's going to be moved to a little bit better, rather than sitting up proud, and as the soil dries a little bit, it'll all crack, and that causes problems for drying of the plant. So there's a pretty successful translocation. Everything is nice and level. The plug fits into the hole that's been cut really well. The only thing that we would do, that we've learnt to do over a number of translocations, is to fill in all the gaps around that plug with washed sand. So that's sterile washed sand. And you have to mark that pretty well, because at the end of the day, that'll just melt into the surrounding soil, and any evidence of the translocation just disappears very, very quickly. Within a year, you can't see that circle anymore. So here's plants that we've moved over a kilometre. These were plants from the Deer Park bypass, and as you can see, they're all sitting in hessian bags. So they've been lifted out of a trailer, out of their supporting basket, and then popped into a hole. So eventually that hessian will rot. It also helps a little bit in that it forms a little bit of a weed mat, I suppose. So as I've said, you've got to mark them pretty well, because very quickly they dissolve into the soil in which you've put them into, and you've got no idea where that plant actually is unless you've marked it very well. You've got to mark them so that you can burn the place. We've been using metal tags rather than little plastic tags or anything like that. Plants can die for a number of reasons, but it's generally drought stress exacerbated by the shock of moving them. We normally inspect plants very regularly earlier on and water them as necessary. And it's easier to provide a watering system. So you can see our, I think it's a 4,000 litre water tank in the background, and we've got a drip system set up. They were planted next to a track, and just for ease of the logistics, we would just hire a water truck to fill up that tank. He could see where the track was. He didn't particularly need any great amount of supervision. It was relatively easy to drive in and out, fill up the tank, which would then just by gravity would just water the plants. Another advantage of picking up plants like this is that you generally, at least for female plants, you're moving the soil stored seed with them. As you can see here, these plants, that's out of a translocated plug. The plants were burnt, and that stimulated germination. And really, to some extent, the only way you can tell that you're getting any level of recruitment is you have to see the seedlings, because if you can't see cotyledons, you don't know exactly where those plants came from, whether it's still the original plant resprouting or something new. So this is the main aim of the game. It's not actually the plants that we're salvaging, it's getting recruitment and getting a new population established. And in the process, we also learn about management of grasslands and the ecology of this species. So in conclusion, translocation should still be seen as a last resort, with avoidance of impacts to plants preferred, but that's not always going to happen. The translocation is potentially to result in a positive conservation outcome compared to the destruction of plants as part of the development. So you're better off moving them than then just being run over by a bulldozer. There's been at least 14 translocations since 1998. In many cases, there's little evidence of stabilisation in mortality, and there's very little recruitment, although we have observed that at 2 sites. And the average survivorship across 11 sites is 33%. Some plants I translocated in 1998 have survived over 20 years and can still be found today. So these plants are still producing seed, so they're making some level of contribution, even though there's no more monitoring to work out exactly what that contribution is. The majority of translocated plants visited show the capacity to produce seed, even though there's very little post-translocation management in some instances. Management techniques, including weed and biomass control and ecological burning, are absolutely critical for these. And as I said, to date, we've been able to manage to set a standard of 5 years for that process. But overall, it's important to involve the recovery team to ensure translocations are well documented and that all the lessons are learned to the best extent possible, so that we can keep learning and keep refining our translocation protocol. Hopefully I didn't go too far over. And I'll now pass over to Rose, who will talk about matted flax lily.
So, good afternoon, everyone. Thank you for the opportunity to present today. I am going to be just talking about a small case study of a matted flax lily translocation program in the north of Melbourne. I've called it A Tale of Two Cities because the program involved one group of plants spread out over 2 different sites, and those sites are a bit different, and that's been a bit interesting for us to see. First, just go into a bit of the biology of matted flax lily and their habitat. Matted flax lily, or Dianella amina, is a tufted mat-forming perennial lily. It is rhizomatous and can form sort of clumpy mats up to 2 to 5 meters wide. The rhizomes are yellow and slender, and they can have shoots 10 to 30 centimeters every 10 to 30 centimeters. Their leaves are a grey-green. I often sometimes get them confused with phalaris leaves in the right light, but they are a bit different colour to that. They often have, or they always have, teeth along the midrib and the edge of the leaf, so they feel quite different to a lot of the other sort of leaves they might look similar to. These leaves are also deciduous in summer, especially if the plants are water stressed or under some other form of stress, so they can die back and then come back in the wetter seasons, depending on what's going on with them. especially if their root systems had time to establish and really pervade the soil. The inflorescence is up to 90 centimeters long, produces these lovely purple flowers, which turn into berries. The flowers come up between October to April, which is when a lot of the targeted searches and annual monitoring for this species tends to occur, because it just makes it a lot easier to find them, especially if they are not especially well marked. This species is widely but patchily distributed throughout Victoria, all the way from southwestern Victoria to the east. The bioregions that it occurs in includes the Victorian volcanic plains, the southeastern coastal plain, the southeastern highlands, and the Victorian Midlands bioregions. You typically find it in grassland or grassy woodland habitats, but they sometimes are found in amongst rocks growing out between the cracks, but they can also just be found anywhere amongst the grass. They're a bit cryptic sometimes. They can appear in well-drained to seasonally wet soils and be in anything from sandy loams to heavy cracking clay. So they can be quite a tough species given certain conditions. Now, we've heard a lot about difficulties and things to consider with translocation programs today, so I'll try and focus this one a bit on Metaphylaxis specifically. One of the things to consider when you're starting off this a program for this species is, what is an individual? The guidelines suggest any plant within sort of 2 meter radius of another plant, they consider that one individual, which is a reasonable guideline, but there's been genetic testing to show that some plants occur all within 1 meter of each other. So what you might think is 1 is actually 3. And then other instances where a plant might be just enormous and something that you might think is 2 or 3 plants is just 1. That is important to work out if you can. Obviously there's limitations given budgets and times, but given the point of translocation programs is trying to conserve genetic diversity, understanding genetic diversity at the start of the program is a key part of it. Another factor you've got to really consider, well, this species is finding the right spot. As I said, they can be quite tough, but if the conditions aren't right, they're not going to survive very well. So while you might be limited, given the development plans or the budget or scope or the timing or a million other reasons, prioritizing picking a good place for these translocated plants is really important, because they just won't survive in some areas, and then that's the entire point of the plan. It's the entire point of what you're doing. Annual monitoring is a key part of any translocation program, and one of the things that is very important to consider and put a bit of work in at the start of a program is working out what data you're going to collect, and then whether that data is going to answer your question about the targets that it's been set. It's useless to collect data that doesn't answer any question about how the program is going and whether the program is going to meet its targets. So it's important to consider that at the start, especially when you think about how a lot of these programs are started off by, one person, one group, but then can be passed along between people and even consultancies. And having data and sort of having it all set up at the start will help ensure that over the life of the program, you're still collecting good, accurate data that reflects on how well the project is actually going. So for Metaphylaxis, I said the leaves are deciduous, especially in summer and heat stress times. That can make it hard to work out if you need to replant this species. Over the life of the program, you will probably need to replant every now and again, considering that all of these plants can struggle to establish, especially if the spot isn't amazing. But you've got to also consider that even if you don't find it in 1 year, you might just be in a bad season, or it might just be a tough season, and it could come up again as soon as it rains or as soon as it's not quite as hot. For this species especially, it comes up from its roots pretty readily, so waiting at least a year or two of it being a no-show in annual monitoring is probably a good idea, considering other factors, just so that you're giving it the best chance to come back. You're not digging it up and killing it, even though it's just happily lying under the dirt. And finally, when you're considering these programs, you've got to set very clearly written out targets so that you can actually measure the success of this program. So having the wording being very clear, being unambiguous, making sure that those targets actually reflect a successful program, like a successful either establishment of the species in a protected area or of maintaining genetic diversity in an area, whatever your specific focus is, making sure that your targets will reflect that. Especially, as I said, programs can be handed from people to people, so it's important to set up targets that people down the line who don't know about project history necessarily will be able to understand and follow. Now, this specific case study, the program started with 48 original plants, which were grown out into 192 clones. These clones were planted into 2 different sites, site A and B. The sites are quite close to each other, within 200 meters, but are a bit different. Site A is slightly higher located in the land. It's between a couple of rocky rises. There's a bit higher percentage of native grasses and forbs. It tends to drain away a lot more. Water doesn't pool in this area compared to Site B, which is lower profile in the landscape. It's a bit of a drainage spot for the surrounding vegetation and urban development. And there's a little bit of a drain sort of right through the middle of the translocated plants, just to give you a bit of context about the 2 different sites. When we look at the replanting rates for this program in the first 6 years, you can see that the first 2 years were quite high, 32 and 55% Site A and Site B respectively, staying pretty high for the first year and then dropping after 2016. In 2016, these sites got an irrigation program, which I expect is what led to that significant drop off. But even after that drop, you can see that Site B has a higher replanting rate than Site A. Anecdotally, we've been able to see that it just holds water a bit too much. The plants don't do as well. The picture in the slide here shows very happy, and well, you can't see its full extent. It's a very large Metaphylaxily. It's got dozens and dozens of ramets that keep coming up every year, and we'd say that was a particularly successful plant, and you don't even get any of those plants in Site B. They're all Site A. As of 2021, so December last year, we've got 76 established clones in Site A compared to only 65 in Site B. In this context, established is being alive for at least 5 years, which is an element of one of the targets for this particular program. No, it's not necessarily significantly different. There is cause to sort of see that maybe Site B wasn't as ideal a spot. And this kind of information isn't necessarily as useful right now for this program, but it's good to know and good to sort of reflect on how well these are going, so that, you know, in the future, if we're developing new programs, we remember and we can learn. So a few of the other, so what we've learned from this program, mostly you've got to be very careful about the selection of sites. Like I said, they can survive really well, but they do need to be in a spot that will support that. So prioritizing that when you're considering all the other factors that come up when you're working on these programs, it's really important. One of the other things we've learned from this is you've got to plant out a lot more clones than you might otherwise think. For this particular program, we planted out 4 clones of each plant, but the success rate relied on 3 clones of each plant surviving for at least 5 years. And in hindsight, that was just being a bit too optimistic about this program. Ideally, we'd probably want to plant at least 6 of each clones to get through, to have 3 surviving, if not more. It just saves a lot of time and protects a lot of the genetic diversity if you start off with a bigger population that you can afford to have random and losses from. That ties to the next one of keeping extra clones in nurseries as the program develops. This is important because no matter how well you set up your program, you probably will need to do replantings over the years. And given the point of this, keeping the genetic diversity of the species or of that area up as much as possible, you want to be able to plant out the clones that have died or the plants that are missing. And so keeping the full cohort of clones as backups in the nursery would be really important for that. One of the other sort of practices that we've found over this project is it's a very good idea to try and forecast throughout the program to see if you're going to be able to meet your targets in a given time frame. So, you know, in year 2 or 3 of your study for working out if in 5 years you're going to have enough plants to meet the targets, you should be working out if that's possible at the moment, if you're needing to be too optimistic about how many plants are going to survive. It's better to replant earlier than later, because for this particular project, if it has to survive at least 5 years, then any planting you do is not going to have any impact on the targets until at least 5 years after that. And given our clients don't necessarily want to actually fund these things to the full extent, got to make it as efficient as possible to make that sell. And sort of finally, you do really need to set high standards for monitoring and reporting. This, you know, for what we've found here, you need to be really clear about what data you're collecting, what you know, how you're reporting on it, and tying that back into the targets, you know, predicting whether or not you're going to meet them sort of at each stage to try and give an indication of how it's going, indicating whether or not any changes need to be made or any updates need to be given to regulators. This is just crucial for any translocation program to maintain their integrity and make sure that they are making the point of what the translocation is for. It could be an interesting area for further analysis, further study, looking into the genetic diversity for this species, especially in small areas. We haven't seen a lot of recruitment from seed specifically. Almost all of it is coming from rhizomes popping up from one plant. So sort of looking into the genetic diversity of the species could be a very interesting area for future work. Thank you very much. Pop any questions in the chat.
Thank you, everybody. Oh, I hope you've got me now. That was really great. Thank you, everyone, for those fascinating case studies and the great images, examples. There was a lot of effort put into those presentations. Warren, Deb, Steve, Rose, questions might be in the chat for you if you haven't answered them already. Sorry, my camera was off then. I hope you've taken away some knowledge. In our final session, we're going to be moving on to some ideas for really optimizing plant translocations, including genetics and what kind of maintenance actually works. But let's have a 8-minute break and we'll get back at 2:45 with Maurizio Rossetto. Thanks, everyone.
This is Microceras scapidura, or a yam daisy. Unlike other species in this series, the roots of yam daisies are edible.
It's hard to describe.
Kind of like a potato.
I can't think of something like a Western vegetable that you could compare it to. It's sort of like a bit like a nutty potato, I suppose.
The term yam daisy refers to 3 different species, all of which have different sized roots, and all are edible, although some may be more delectable than others. Here in Melbourne, the Wurundjeri Woiwurrung people refer to the yam daisy as murnong.
It was a large staple food for southern Aboriginal people and, yeah, of incredible cultural importance.
Sadly, it is not known if murnong refers to Microceras scapidura, another species, or all 3 species of the yam daisy.
That's unfortunately knowledge that's been lost.
Today, Microceras scapidura clings on in only 3 known populations amongst the urbanising critically endangered grasslands surrounding Melbourne. Keeping this species alive requires ongoing management and translocation. For 30 years, the Merri Creek Management Committee, originally led by Brian Bainbridge and now Michael Longmore, have been trying to create self-sustaining populations through translocation of Microceras scapidura, but their attempts had largely failed.
We planted 20 one year and we got 1. We planted 200 the next year and we got none surviving.
Plants were originally grown in tube stock, which is a common method for translocation, but it meant that the tubers hadn't properly developed to withstand the hot, dry summers and cracking clays.
So they might live for a year or so, flower quite well, but then nothing would emerge the following year.
During the millennial drought, we actually witnessed the disappearance of one of those populations. So we're worried about losing the whole species.
Brian and Michael both spent considerable time trying to learn how to grow the species, and it was only really by luck that they stumbled on a better approach.
Where they had survived, it was seed that had fallen from those plants. We got germinants, and then those germinants appeared to survive for a much more extended period of time. So that gave us a hint that direct sowing might be something worth pursuing.
As the plant grows from seed, the roots find the natural cracks in the heavy soil, and the tuber grows in the right place. But getting a plant to survive is only part of the process. It needs to reproduce and recruit.
Fire was incredibly important to the landscape in maintaining and managing the land. So yeah, after you burn an area, you will, if there's murnong there, you will find them popping up.
It also opens up space amongst the grass tussocks where seeds can fall and germinate.
There was cultural significance involved with the collecting of yam daisies, which was usually done by the women with their digging sticks. That disturbance of the soil and aerating the soil also helped to create space for regrowth and for more murnong to grow.
Also, small mammals that used to exist in the landscape and would go around nosing through small areas, turning over small amounts of soil.
So when they brought in the sheep and the cows, which trod down all the ground and made it very difficult for it to grow back because of the compacted soils.
What the team discovered is what Wurundjeri had known for thousands of generations. Microceras scapigera relies on fire and soil disturbance, or digging, to promote germination and growth of new tubers.
We hadn't spent the time listening to find out what was needed.
Brian knows that historically, Merri Creek Conservation Committee and Wurundjeri weren't always working on the same timelines and with the same objectives.
Our organisation, we had particular goals we wanted to get out, and we need to align those to get the richness of what happened. And when we did get things aligned, when we have had things aligned, we've had so much come back to us as well.
Today, the Merri Creek Management Committee, Friends of Merri Creek and Wurundjeri Woiwurrung are working together to conserve the species and the grasslands.
In combination with Western science and traditional knowledge, using that combined knowledge is to get the best benefits for the environment, for Aboriginal people, for everyone really.
Wurundjeri are also in collaboration with Joe's Market Garden in Melbourne and have taken initial steps for cultural production of the species.
We're growing murnong up there to increase seed stock and help revive the plant because it is endangered.
3 populations persist. They are not self-sustaining, but they are not going to go extinct. When traditional knowledge and Western science work in collaboration, the benefits are rich for the species and all involved.
I would encourage anyone that's working in areas that have an Indigenous aspect to it to contact their traditional owners and start a relationship.
While it hasn't been done the right way in the past, it is never too late to start. The Murnong story shows us this.
Okay, we are back for session 3. This is our last session. We've got some really great advice coming up next about some new, maybe not so new ideas for maximising efficacy of our translocations. First up, we have Professor Maurizio Rossetto from the Royal Botanic Gardens in Sydney talking about conservation genomics for optimising collection and population design. And then Professor Peter Vesk from the University of Melbourne is going to be building on some of the work that Dr. Jen Silcock mentioned this morning, and some trends in maintenance about what's working and what might not be worth your time. And then we're launching the last video in our series. It's about orchids. You'll notice that there's no orchid people here today, and that's because it's prime orchid translocation season, very poorly timed by us. And finally, finishing up, Dr. Amelia Martin-Jensen from the ANPC is going to remind us that sometimes translocation is not the best option and give us some ideas for alternate conservation methods. Okay, thank you so much. Maurizio, over
Thank
to
you,
you.
Sean, and thank you for inviting me to participate to this. Hopefully you can see the screen. So my name is Maurizio Rossetto, and I head the Research Center for Ecosystem Resilience at the Botanic Gardens in Sydney. And what we do a lot of conservation and restoration genetic work. And so what I'm hoping to do today is to remind you all how important genetics are for supporting your translocation and your restoration projects. And also that, and I think some people mentioned it already, how it's important to start from a genetic when possible, or really should always be done, but to start from the genetic, from a genetically informed position. So one of the main reasons why we need to think about genetics is that now that we understand more all the processes involved in ecological restoration, the translocation of threatened species, hopefully we're no longer focusing just on quantities, on putting down 1,000 trees or 1,000 plants, but on quality as well. We want those 1,000 trees or those 1,000 herbs to establish self-sustaining populations that will be resilient to future changes. And unfortunately, the reality is that to be able to do that, we need to have some sort of understanding of the genetics of the species. And I don't necessarily want to go into the details on why we need to do so, but let's just say that now, whilst in the past trying to get the necessary genetic information to support your translocation programs was complex, costly, difficult, it would take a long time, the genomic era has really changed that. Now we can really access a lot of information, much more information than in the past, and this can be done in a easy to access, quick and affordable way. In fact, one of the things that I really want to remind you is that if you need genetics, you do not need to have a genetic lab. Nowadays, it's really easy to outsource stuff. In fact, all of the work that I will be talking about, we outsource. We don't actually do it in our labs. We get all the sequencing done, outsource all our sequencing, and then we can focus on analyzing and interpreting the data, which is really the more interesting thing part of it all. Another important thing to consider is scaling up, right? If you consider economy of scale, it's where you really can do more. You can really access more. You can really do more and obtain more data and be able to interpret it better. Right? And I'm not saying that it's not important to focus on single species, but really we're in a time now when we need to think bigger. And so to be able to do that, then we need to develop workflows that will help you to do that, will help you to work with multiple species. And these workflows are important because they really provide you with a data set that will help you analyze, satisfy multiple purposes. So what I've got here is it's kind of a representation of what a workflow, the type of workflow. I mean, let's be frank, it took us quite a few years to develop the perfect workflow, but now it's a workflow that we can apply to all sorts of studies. What this workflow needs to be done is to simultaneously sample as many species as possible, as many species as relevant, across their entire distribution in a way where you will have comparable data sets. To do that, to collect all the relevant information, including the metadata, in an efficient, replicated, consistent way. We use an app for that, so it's now super easy. And we have a specifically developed database to keep all that information. So not only the sampling information, the collection information, but the genetic information, all the additional metadata, and then make sure that all this material also well stored so that if we need to do additional analysis, it's easy to reach, or if someone else is interested, we can pass it on. Once we've got all of that, then we can do our with one single genetic sequencing event, we can really have all sorts of options of what we can do with that data. And I'm not only talking about scientific papers exploring the dynamics or evolution of single species, or bringing together multiple studies to look at biogeographic processes or adaptive landscapes. We can really have very, very applied outcomes, all from the one specific data set. We can guide the establishment of seed production area, which is such an important strategy now in restoration and translocation. We can guide the establishment of existing collections, be seed collection or horticultural collection. And more relevant to what we're talking about today, we can directly support threatened species translocation and resilient ecological restoration practices. So with that in mind, I want to start to talk about threatened species, and then we'll talk about common species and common species restoration. We published last year a simple workflow on how to develop genomic studies for threatened species. And although flowcharts always kind of look complex, this really is very simple. All it relies on is a carefully planned, simple initial sampling strategy. And this can be done at the initial, when you start your project, right? It can be done when you do your first survey. You sample all the material, even if you don't know if you can use it or not straight away, you can store it away indefinitely and use it later. So it's not a question of complexity there. It can really overlap with other activities as part of the project. Once you've got your material, one simple collection event, then you have one simple sequencing event. So you sequence all the data, all the samples that you need, and then you can put it through a whole range of different analysis to answer all the questions that you might have in relation to threatened species management, including very, very applied ones. So when we produce, for example, when we work with threatened species and we produce the report, we answer all the questions with very, very practical outcomes, with very practical guidelines. And I'll give you a few examples in a minute. And of course, also all those practical guidelines, in particular relating to translocation, are backed by methodologies that we've developed through the years. And this is some of the example of the work that Jason Bragg has been leading as part of our team. So we have developed very specific ways to analyze the data to ensure that we get the outcomes that we are after. So let's have a look at some of the examples of what we can do with this single one data set. And one of the examples I want to use, and this is work that led by Samantha Yap. Sam coordinates all our SOS, all our threatened species programs. This is an example of a rainforest tree, Daphnaandra johnsonii. It's mostly distributed south of Sydney, and it's a typical situation where you've got small fragmented populations, and most of those populations are infertile. And the management team were interested in establishing ex situ collection. In fact, they did establish ex situ collections to be used in the future in translocation. So we wanted to make sure, amongst many other questions that we had and we answered about the species, but we want to have an understanding of the distribution of diversity. We want to validate their existing collections, get an understanding of what they already had, and trying to optimize a strategy that would maximize genetic diversity and most likely restart successful reproduction. Because from experience that we worked on many other similar species, the main reason why populations were not fertile is likely to be that they were highly inbred, and most of those rainforest trees being preferential crosses, that would cause this lack of fertility. Anyway, let's have a quick look at this graph on the right here. And what this is it gives us an idea of the kinship, the relationship between all the individuals within and between populations. So what we've got on X and Y axis are the single individuals, and here on the diagonal is a representation in color of the kinship, the relatedness between pairwise comparison of all the individuals. If the color is red, it means that they are identical. If it's orange or dark yellow, it means that they have this individual are closely related, and that this cream color here tells us that there is no close relatedness. To cut a long story short, what this is telling us is that although the populations are genetically distinct, sufficiently genetically distinct, within population we have high levels of clonality, and in fact some of the clones extended in a linear fashion to over 800 meters. So it was a little bit surprising, a bit more than what we expected. But generally, most of the populations are either clonal or highly inbred. So that explains straight away why sexual reproduction was failing across nature within those populations, and also suggests and support the idea of mixing those populations to kickstart natural processes again. What we just briefly, the other thing that we were able to do with the information was to assess their existing ex situ collections. And so, for instance, we can see that some of the collections were just replicating the single genets, so they had multiple ramets, single genets, which is a bit of a shame, but at least now they can rationalize the collections. On the positive side, we could identify that some of the genets in the ex situ collection were unique, so they were no longer present in the wild, which is again one of the great things about ex situ collections. And also we could identify some of the individuals that were of unknown origins. So from there, then you have the information on what the genetic diversity is across all those various sites. How do you use it for translocation? Well, what you can do there is what we developed is a method that uses a simulated annealing algorithm to maximize diversity and minimize kinship for any given number of individuals. So if you want to plant back 40 individuals, this is what you need to do. These are the ones that you need to use. This is the ones that you need to propagate. If you want to plant 50, this is what you need to propagate. And this is because obviously not all individuals are equal. There are some that contribute more to the genetic diversity. So if your objective is to maximize genetic diversity, then we can design that very specifically, to the point that we can then design the planting strategy as well to minimize relatedness between single individuals. An important thing about all of this, of course, is that we can give you all the indications and guidance in the world, but the horticultural realities might be very, very different. So if there are issues with that, we can just reanalyze the data and find the next best solution. So it's a very, very practical outcome, and this is something that we do as a standard in the projects that we work with. Of course, we can take it one step further and look for more complex solution. So for instance, you might have competing objectives that you want to achieve in your translocation. For example, you might want to bring in some climate-ready material, some future capable material, or as in the case here, some resistant material. Now, you don't want to limit your translocation to material that is future ready or material that is resistant, because that's one issue that the plant has. There are many, many other issues, and you don't want your translocation to be just adapted to that specific trait, a specific physiological response. You want your population to also be adaptable to multiple other potential challenges, selective challenges. So, for example, here in the case of rhodamnion, this is work led by Stephanie Chen. We want to make sure that we had identified some resistance, some individuals that are resistant to rust, but we want to make sure that we could mix it with the broader diversity of the species in order to have an adapted and adaptable population. And again, to do that, there's we developed a specific multi-objective optimization algorithm where you can balance and decide how much of each of the two competing objectives you might have. So you want to ensure that it's sufficiently diverse and sufficiently adaptive. Anyway, the point of this is that what I'm trying to show is that with a single, with the one single data set, you really can provide a whole lot of solutions. And we now, since 2018, mid-2018, we've started work on more than 50 species, and we've finished most of them, and we've got another 20 coming online in this financial year. So it can really be done efficiently. Usually one team member will work on 10, 15 species in a year. And once we have the data, really it takes 1 to 2 months to do the sequencing and 1 to 2 months to do the analysis. So again, if you work with multiple species, you can work on them all together and be much more efficient. Again, the point of maximizing and multiplying efforts. Obviously, we don't only work, and I know now I'm quickly running out of time, but we don't only work on threatened species. We really started to develop all those strategies for common species, and common and threatened species have got slightly different ways of dealing with the data, because with threatened species we can target the single individuals. With common species we tend to target populations, locations, provenances. I don't particularly like that word, but let's call it that, genetic neighborhoods. And really we started all of this process with the Restore and Renew project. That's how we developed all those workflows. And the objective there was for us to provide freely accessible information on the distribution of genetic provenances, the distribution of genetic diversity of single species, and how that related to climate. So if you go onto the web tool, what you can do is you can select your site and your species of interest, and what the web tool will give you is an idea of what the local genetic provenance is and what the various climatic conditions, future or current climatic conditions are. So you can make decisions on how to sample according to provenance. We've got data on well over 120 species, and although they're not all on the web tool yet, we are continuously expanding now with more expanding. We initially started in New South Wales, and we're expanding in Queensland and a little bit in Victoria as well. But really what we are doing now is expanding the type of information and tools that we can provide through the web. So for example, in the absence of genetic data, in the past they've developed a number of really interesting generalized guidelines which provided scenarios on how to sample material for those specific scenarios. If you want to climate proof it, if you want to local provenances, if you want to admix, etc., etc. And this is one of the most popular one of the best ones, and a set of scenarios published by Super over a few years back. Reality is that for those scenarios to really act on those scenarios, we really need the climate and environmental data. And so we are now working on developing methods that are easy to develop and easy to interpret to actually provide that type of information on the web. And this is a PhD project that Richard Dimon, that many of you might know, has just started. So what Richard is working on is on a small set of rainforest species. He's working with about 20 or so species, and then we'll probably add in the next year another 30 or so that have been sampled across their entire distribution. And so that we can then use all those genetic data sets to kind of develop these strategies. So for instance, if we want to go and try to maximize, and I should say this is a project that we do in collaboration with Big Scrub Land Care Group up in the Northern River. So they were interested in establishing both seed production areas and being able to source material from the local rainforest to restore cleared areas. And so the first step there was to look at across all the species, as I mentioned, sample across their entire distribution and get an understanding of the distribution of genetic diversity. So that's what we've got here. On the rows, we've got the single species. On the columns, we've got the individuals in the population. Again, you can see how it's important to sample from the same locations. We've got very, very replicated and comparable data sets. And the colors identify the provenances, the genetic groupings. And the ones that we're interested in is in the northern rivers, the red colors in the northern rivers. Once we have the information, we can use resampling models and simulation to identify a minimum number of populations and individuals that are needed to reach a maximum of diversity, whatever that might be. So here in this case, we look at representation of 90% of allelic diversity, local allelic diversity. It's just a measure. There could be other measures as well. So we do that for every single species. And once we've got that number, we do pairwise comparison amongst all the population to get an idea of how disjunct they are, so that then we can we have a population that are best suited to sample for you to maximize diversity. Once we've got that information for all the species, then we've got a very nice way, and so the population, these are the populations, these are the species around here. Once we've got a good understanding of that, the green identify what are the best population to sample. And we can then also look at the practicality of sampling, ensuring that we can access as many species per site, so that we really rationalize the sampling strategy. So now we've got very, very specific guidelines on how to sample 20 species within a whole region to maximize diversity. If we want to look at another scenario, for example, future proofing, we can look at environmental models, identify where the species are already experiencing the condition that your restoration site will experience in 2070. We can then impose those populations, a relative contribution from those populations, repeat the exercise, and so on. And of course, we can repeat that for any other type of scenarios. So again, those are very practical guidance on how to sample to achieve specific objectives that you might have in whatever restoration scenarios you are interested in. Let me quickly bring it in, because I'm aware that I've wasted a bit of time trying to reconnect. Take-home message here is that genetics are important, and they really should be considered as the starting point of any translocation and restoration process, because the amount of information that you can get for them is really phenomenal and can guide all sorts of processes and component of your management and restoration processes. And obtaining this information, especially if we think about the scaled up workflow, is really much more accessible than it was, and much more affordable than it was in the past. And no one needs to be an expert. There's always opportunity to collaborate or to outsource some of the work externally. A last couple of important points I want to make. In the past, I always used to say, we do not have time to do genetics. Absolute nonsense. As I mentioned before, you could sample your material in the first survey that you do. Once you've done it, and I hope that when you're dealing with certain species, you do surveys. Once you've done that, then the material can be stored away, and that's it. It's all about then finding the support and the money to do the genetics, but really you're not holding back any of the other processes. And this is valid also from a scientific point of view. There's always time, there's always time to write papers later. We can provide the priority is to provide the guidelines for the management of those species. And having lots of data, of course, will make much better papers anyway. All right, I will leave it at that. I hope I didn't delay too much. Thank you all for your attention. Again, apologies about the technical issues. Please don't hesitate to get in touch if you're interested in any of this, or if you want any support, help, or collaborations. And of course, before I want to acknowledge all the traditional owners of the many lands that we are working on, have been working on, will be working on in the future, as well as our funding and research partners, but more importantly, of course, the research team, that the ones that are doing most of the work that I've presented here. Thank you.
All right. Thank you very much for having me along here. Thank you, Chantelle, Joe, everyone for organising this. And I'd like to apologise for what look like very ambitious titles. I don't think they're pretty awful. I'd secondly like to say, practitioners, all of you wonderful people who pay very close attention to the work that you do, blur your eyes. This is a really, really coarse analysis that I'm going to produce. It's a short talk and won't go into any near the level of detail of Maurizio or others like this. This is work on behalf of a group of people here. You can see them listed up there. It is building upon Jen Silcock's fab work in synthesizing the results of the terrific work of people in this network and beyond. So respect to you all. Also, Laura Simmons. And Mike Whitehead has been driving this analysis while I cheer from the sidelines. But Mike has a new day job. He's working for the Victorian state government on plant invasions rather than conservation. So it's fallen to me to speak, and so you have to put up with me. I apologise. The other thing I need to say here is that this is, essentially an outcome of the Plant Translocation Working Group funded through NESP, Threatened Species Recovery Hub, and Project 4.3. Okay. So in translocations broadly, I would say that our aim is to restore viable populations. We may be establishing new populations or augmenting existing populations, but we want those populations to be viable. And I'm an ecologist rather than a geneticist, and I think about the ecological population dynamics. And in this context, my thinking is about indicators like the extant population size. So how many plants are in the population. And that's an indicator that we're going to use, viability, and I'm going to speak to in this data set, in this analysis and talk. Another indicator may be recruitment, so the appearance of second or subsequent generations. And so in this talk, I'm not talking at the level of an individual seedling, an individual germinate, but the observation of some recruitment of that population. Okay. Now, ideally, as many people have mentioned, we would dearly love the information on survival of individuals, on their growth, on their fecundity, on a seedling establishment and the like. And Leonie Monks has done some terrific work in getting into this finer grain of detail. Perhaps she's going to talk about that tomorrow. But this is not available to us at this broad level of this continent-wide analysis. Okay, so we're going to be talking about extant population size and recruitment. So Jen talked about the translocation data set, which dealt with 1,001 translocations that she managed to get into the database. We filtered this down to those that we could get actions for and ended up in it with a data set of 665 translocations. And you can see there on the map how they're distributed across the country. Some in the tropics, down the east coast through Victoria into South Australia and the southwest corner, a bit in Tasmania, but broadly not in the dry and top end. Okay, so because of that, it's spanning diverse vegetation types, diverse environments with diverse life forms, things from rainforest to semi-arid grasslands, trees and cycads through to annual forbs and geophytes, and diverse taxa. Now, in these analyses, we're going to try and get an overview, but be aware that it's going to have all of these this diversity underpinning it, which leads to a lot of noise in the data set, if you like. So in the analysis that Jen did in that paper, there was in the interviews with practitioners, there was this synthesis where she attempted to identify those factors that practitioners identified as contributing to the success or failure of their translocation. So the white filled bars indicate the number of translocations that practitioners said the factor contributed to success, and the hollow black ones are failure. And I'm going to highlight 4 things there, because some of the things in that graph you'll see are not really actions so much, but other sorts of factors. Okay, so it's like species biology. It's not really an action. In this, you can see that there was an attribution of the importance of weeding. Weeding's fallen underneath that blue here. Grazing or trampling, watering, disturbance management were all identified as things that practitioners thought were important. You can see they also said that the lack of maintenance and long-term something or other was a contributing factor to the failure. So practitioners recognize that getting jobs right matters. And sorry, I'm talking as an academic. I don't do anything. I just wax lyrical. Okay, so these are the 4 sorts of actions I'm going to be speaking about. We were able to extract information or attribute actions as along this classification. And amongst those 665 translocations, there were 44% of cases where there was watering after planting. 2/3 roughly, there was some sort of grazing protection. In roughly 40 to 45%, there was either slashing or weeding before or after the planting. And then in a very small number of cases, there was burning. And of those cases that were burning, they were predominantly planned burns rather than wildfires. Okay, so the important thing about those numbers is that the first 4 of these give you a good balance of presence or absence of the management action, so they enable us to analyse these. Rather less information about burning, and when you have less information, it means your uncertainty analyses go up. The hokey kind of icons I've made here indicate watering here, caging to protect from grazing, and burning down here. You can see that there's no icon for the slashing or weeding, and there's a clue in that that broadly in my analysis, in our analysis, sorry, these weren't all that important. Okay, so the sad fact of the matter is all things are mortal, plants die, and so eventually populations will decline, okay? Numbers will decrease when we introduce a cohort into a landscape. Unless, of course, recruitment happens. If we have the happy event of recruitment, then potentially the numbers may increase. So what we have on the right-hand side is the output, or what we call a coefficient plot from a generalized linear mixed model. So it's a glorified regression model where each of these factors on the left-hand side are entered into the model, and they have effects, if they are on the right hand of 0, then they are positive effects on the outcome, and if they're on the left-hand side, they have a negative effect. So this is on the number of plants surviving at the latest census that we had, okay? So years here is years since the event translocation. It's negative, meaning that over time, plant populations decline. Unless, of course, if you've got recruitment over here. And you can see that recruitment effect is positive. It's much larger in magnitude than the years. And there's also an interaction between years and recruitment, which I'll display in a couple of slides to show how to interpret that in a projection. In the yellow, though, is the overwhelming effect of the number planted. It is overwhelmingly the most important thing to do. Plant as many plants as you can to keep as many plants into the future. Outside of that, our actions are up towards the top. And you can see here, grazing protection has a positive and quite certain effect. So protecting from herbivory is beneficial by and large. We see almost no effects of supplemental watering or control of weeds through weeding and slashing. The mean effects are close to zero and the uncertainty spans a wide range. Okay, so it seems that the important thing is plant a lot, protect from grazing, and we want to get recruitment. Oh, this slide just shows that the ultimate success does vary a lot between species, between different life forms here, and between different vegetation types. You can see that there's variation in where the point estimates lie, but the uncertainty overwhelms this. What this means is that when it gets down to individual vegetation types or individual life forms, we don't have that much information, okay, at this level of a continent-wide analysis. So getting back to projections, here's the coefficient plot in the top right. Each of these 4 panels here are combinations of the expected population trajectory over time under recruitment, no recruitment or recruitment, and no grazing protection or grazing protection. So the top left is the situation, the base case of we have no recruitment and we have no grazing protection. And an initial cohort of 100 propagules leads to the sad situation that in 100 years, we have less than 5 individuals persisting. Now, this is for the base case of perennial forbs. There are those extra effects of growth forms, but the slope of the line wouldn't change. The y-axis is on a log scale, so it's in orders of magnitude, 10 times in each of those. So you can see there's a precipitous decline. Unless, of course, we get recruitment, in which case the slope of the line is reversed and we actually get the happy situation that numbers increase. They don't just stay stable, but they actually increase if we observe recruitment. And if we then have grazing protection, what you get is a little shift up or down on the slopes of those slopes. So here, the situation is that the trajectory ends at 5 rather than under 5. And in this case, it ends a little bit higher than in the no grazing protection. But it's within the bounds of uncertainty. Grazing protection doesn't matter as much as do you get recruitment. So clearly then, how do we get recruitment? Well, clearly we need to get pollination, we need to get seed set, we need to get dispersal, effective germination and seedling establishment, none of which we know about in this analysis. All we know about is, did the population recruit? So that's a 0 or a 1 in this data set. So it's probabilistic, and we have to do a different analysis here, which is like a logistic regression. Again, we have a coefficient plot on the right-hand side with effects on the right-hand side being positive and then on the left being negative. So time matters. Years now has a positive effect. It says that we need to wait for recruitment to happen. The longer we watch, the more likely we are to observe recruitment within the population. And numbers matter. Again, the greater the number of the translocated population, the greater the likelihood we achieve recruitment, okay? Of the management actions, this time grazing protection has no effect, though burning does. In those small number of situations where burning makes sense for stimulating recruitment, it has been shown to be effective. There's not much visible effect of weeding control, and watering control, it might have a positive effect, is unlikely to have a negative effect, let's put it that way. So the key thing here again is plant more. Illustrating the trajectories here, again, the coefficient plot up on the top left, top right, and the 4 combinations of no burning on the left, burning on the right, no watering on the top, watering on the bottom. So in the base case at the top, at 20 years, our population would have a 50% chance of recruitment. At 0 years, it has no chance of reproducing of recruitment. And at 40 years, this population is quite likely to have achieved recruitment. If we then water, at 20 years, that probability is up to 62%. So we've improved the probability. And in those cases where burning makes sense, it improved up to over 80%. And the combination of burning and watering led to the probability of being over 90%. So those combinations of management effects were incredibly important in the analysis of what led to recruitment. And this is just showing that there was some small effects there was variation amongst all of these things, and a very small bit of information that suggests that potentially some management effects differ between ecosystems, as you would expect them to. Burning makes no effect in rainforests. Slashing and weeding might have differed between semi-arid grasslands and shrublands, okay? But by and large, they were weak effects, and the overall effects were pretty consistent. So how do we bring this together? So this hokey little cartoon is my way of synthesizing the two analyses here. We have in these things up here, the number surviving, the number planted, recruitment. We have arrows between them, red ones being positive, blue ones being negative. So to get more surviving plants, we plant more, because over time, those numbers decrease. Unless, of course, we get recruitment, in which case the numbers increase. Grazing protection will benefit the numbers, but probably also helps to keep the numbers up so as to achieve recruitment. Burning only was seen to stimulate recruitment, and watering may have improved the chances of recruitment. And if it did, it was probably through keeping the numbers of individual plants up so that so that it was a greater number to have a greater chance of recruitment. Little evidence of weeding effects again, I would say. So please, to all of you who do these sorts of activities, please set up to monitor and go back and do that monitoring. If nothing else, we need to know numbers. How many went in the ground and how many were censused at some time into the future. The graph on the right shows from the Silcock et al. Paper that in that database, the time since the translocation was achieved, and that the great majority of them are 10 or years or younger. 10 or less years in age. So we need to watch them for longer. The probability of recruitment, you might remember, only exceeded 0.5 after 20 years. So we need a lot more systems that we watch for longer. Clearly, more than that would be useful. Did they survive for 1 year? Did they reach flowering? Was there recruitment? All of these things are important for us to learn from. And remember, in interpreting what I've been saying, these are mostly not experimental. What Jen said was these are experimental, yes, but most of the translocations are not set up as randomized stratified experiments. Often the actions are applied because practitioners believe they stood a chance of working. Especially think of supplemental watering. Typically, you will only water at a place and a time when you think it's going to be needed. You're not going to apply water if it was a wet year in a wet place. And so we won't see the signal that says, well, watering actually matters a lot. It will be more that, well, watering mattered in those times and places where it was needed. Okay, so thanks to those institutions. I'm happy to take any questions in the chat, and I'll stop sharing.
Today, we're talking about orchids. That plant family that has captivated naturalists since Charles Darwin.
Orchidaceae is a large plant family. There's still well over 25,000 species globally, and they come in every colour, form and size that you could think of.
Orchids have a reputation for being one of the most difficult plant groups to propagate and return to the wild, because of some very unique requirements. The first is habitat. Phellimitra epipactoides, or the metallic sun orchid, occurs in a range of habitats across Victoria, Australia. But in East Gippsland, it has a very specific habitat.
It occurs in an ecotone between saline wetlands grading into Banksia woodlands. And even within that ecotone, it occurs on slight rises. So the places where it did occur was really limited.
On top of that, the wild population was facing many threats.
It's a highly palatable, highly visible species. The population were being overrun by deer.
The wild population was well known to naturalists, all wanting to look at Australia's largest sun orchid, but sadly trampling it in the process. This niche habitat made it incredibly challenging to find a suitable planting location for a conservation population. That was until Gippsland Water volunteered.
They had this ecotone. How lucky. And they said, yeah, you can plant there. And yeah, we'll build the fence.
So we had to set up an enclosure, excluding all the sorts of things that like to eat orchids on occasion. We put in 192, I think we put in. And in 2019, we had 165 plants re-emerge. And in 2020, we had 162 plants re-emerge. The next achievement we'd like to see is recruitment.
Even though the team were lucky in finding the right location, it still took around 22 years of surveys and research to plan this project. And this is because where to put orchids is only one part of the puzzle. Orchids have very specialised propagation and pollination requirements. Dr. Nushka Reiter has experience of over 50 orchid translocations and has been world leading, unravelling the mysteries of this plant family.
Basically, all of our terrestrial orchids are reliant on mycorrhizal fungi in order to germinate in the wild.
Mycorrhizal fungi live within and around the roots of most plants, providing mutual benefits of food and chemical signalling. But for orchids, these fungi are highly specialised. And the seeds do not have their own food reserve, known as endosperm. Instead, they rely on fungi for energy to germinate and grow.
Providing that the mycorrhizal fungi wasn't there, then they wouldn't survive and they wouldn't be able to recruit.
One of the species that Nushka has provided groundbreaking research on is Callidenia colorata.
It's a gorgeous little spider orchid. It's nationally endangered, and we were able to discover that there wasn't that many species that they associated with. Having identified our mycorrhizal fungi, we'll then use that in the propagation of the species.
Nushka and the team also looked at the distribution of this mycorrhizal fungi across southern Australia.
So there's lots of other Callidenia species out there that are more common that in some cases share the mycorrhizal fungi with these rare species. It's really helpful to have indicator species when you're choosing your introduction sites.
An orchid's relationship with its pollinator or pollinators is critical to the orchid survival.
Orchids have some of the most amazing pollination systems in our plant kingdom.
Orchids use a wide variety of complex, highly specialised methods to attract and select the right pollinator. This can be through simple mechanics, the pollinator being the right size for the orchid, or it can be more complicated, such as sexual mimicry, food deception, and food rewards.
Often when you have these food rewarding systems, there might be several pollinators involved, or it might not be specific. Caladinia colorata was actually the first proof of a food rewarding but specific pollinator relationship. It was just one thin-eyed wasp that is the right size and the right fit. And this orchid provides a really small amount of nectar on the labellum.
That's right, there's only one species of insect that pollinates this orchid. And so this has huge implications for when you are thinking about translocations.
So you really need to make sure that the pollinator is present and that you're getting your plants pollinated before you introduce them in large numbers.
The detail and attention given to orchid translocations is yielding dividends. They get this attention because the project would fail without it.
The populations are self-sustaining, so there are now more recruits than there were plants that were put in there originally.
But there's one key thing we haven't addressed. It seems like a huge amount of time, cost, and labour for such a niche species with niche relationships. Why not let them go extinct?
Orchids represent 16 to 70% of our nationally threatened flora, and so there are a large proportion of our threatened flora. Having a specialised program that helps deal with some of the intricacies of that is really helpful. However, the lessons learned are really applicable to many other threatened flora. It really hasn't been embraced across translocations globally, and it requires quite a lot of work. Many of our Australian flora have specific pollinator associations. The majority of our other plants do have mycorrhizal associations, and there's been lots of research that has shown that with their mycorrhizal fungi, they are healthier, more vigorous plants in the wild.
If we want to have the best chance of saving threatened flora from extinction, we need to learn from these orchid translocations and understand it takes years of research, planning, and dedication in order to successfully save plant species.
Be all good. So before I begin, I'd like to acknowledge the traditional owners of the land where I live and work, the Darug and the Darawal people, and I pay my respects to their elders, past, present, and emerging. Today, we're going to talk about alternatives to translocation when moving, planting, or restoring isn't the only choice. And I think some of the content you'll see today harks back to Lucy's talk at the beginning of the session. And congrats to Chantelle and the team in the background there for a wonderful lot of presentations today. Thanks to all the presenters. So the information that I'm sharing today is based on the new edition of the Germplasm Guidelines, as well as information in the Translocation Guidelines, which were published in 2018. There's video playlists for both these topics on the ANPC YouTube channel. I'll also briefly mention the Flora Bank Guidelines for seed-based ecological restoration, which was published last year, and all these guidelines are available for free download via links on the ANPC website. So you might start to consider a whole range of strategies for plant conservation during the recovery planning process or when you're looking at your target species. In the Translocation Guidelines, Chapter 2, and there's a screenshot here, deals with decision making. So is translocation a feasible and effective option at this time? Assisted conservation measures such as translocation exist alongside a whole suite of management options for conservation of species and ecological communities. And the same is true for ex situ conservation, which is conservation of plant material away from the natural habitat. Translocation and ex situ conservation are only used when they're necessary, and the maintenance of genetic diversity is important to consider, as you've heard earlier today. We put together this infographic when we were working on the germplasm and flora bank guidelines, and you can see the management actions for in situ populations or natural habitat here in green. Depending on site conditions, the management actions include maintaining viable habitat, promoting natural regeneration, assisting regeneration, and more intensive activities under the umbrella of translocation or restoration, such as seed and plant collection, direct seeding and planting. So translocation is, of course, part of the bigger picture of management of target species and communities. And then you can see ex situ or off-site collections provide both information and a source of propagules to support translocation and other in situ conservation actions. There's a range of collection types in the grey box here. So cryo storage, seed banking, micropropagation or tissue culture, and living collections. And these rely on techniques such as seed germination or nursery propagation for activation. And you've heard some great talks today from Meg and Warren on these topics. The capture of genetic variation, the scale of the seed collection, and the time frame involved depends on the conservation goals for the species or the plant community that you're working with. So now we'll consider some of the ex situ alternatives if you're planning to move towards translocation in the future, or if translocation is not an option at this time. They'll provide you with both the plant material and information that can be used to improve translocation success as you observe the plants and understand their management and perhaps their breeding system and genetic diversity over time. First, a quick note about terminology. Germplasm is a term for any type of plant tissue that can be turned into new plants, including seeds, cuttings, spores, and pollen. In the guidelines, we have chapters on seed banking, tissue culture, cryopreservation, and living collections, which are all germplasm collections that make it possible to capture genetic diversity for the future. The germplasm guidelines begins with an introduction and then a chapter outlining ex situ options available, what to consider and how to prepare for germplasm conservation. So this complements the information that's available in the Translocation Guidelines, particularly chapter 6. Throughout the guidelines, we've included graphics such as flowcharts, pop-out boxes, and references to source documents to bring together lots of information in a way that's easy to use. Now, you don't need to see all the detail in the flowchart here, but notice that there are lots of things to consider before you make a seed or a cutting collection for conservation purposes. And I've copied some of the key considerations here in the dot points. Is there a clear link to the recovery process? Is it a precautionary collection? Is it an exceptional species and somewhat difficult to store? What's the time frame available? And do you have the necessary resources, permissions, and permits in place? If you're new to the ex situ conservation process, talking it through with an experienced staff member at a botanic garden and the plant experts in your state's threatened species unit is a really great way to get up to speed. Once you've decided to make a collection, we have a few chapters with key information. We've got a new chapter with genetic guidelines for acquiring and maintaining collections, with options based on whether you have genetic information for the target species or not. We've updated the chapter on collection planning and techniques with checklists, hygiene tips, guidance on when to collect, and photos of different collection methods. And Meg gave a great overview of the collection process earlier. Note that it can be important to make more than one collection trip to get the quantity or diversity of germplasm you need to meet your conservation goals. We also have a new chapter on identifying and conserving non-orthodox species, such as some rainforest species that can be difficult, that can't be dried or frozen and maybe can't be stored long term. And sometimes we call these exceptional species. Often these need to be conserved as living collections or using a combination of tissue culture and cryo storage. And if this interests you, there's plenty of additional information, including videos available through our YouTube channel. In most cases, seeds are the lowest cost and easiest collection type to maintain, taking up very little space. Although many Australian species have long-lived seeds, we have to remember that seeds don't last forever. So we need to find a way to use them and to regenerate or recollect that species in the future. The guidelines have updated chapters on seed banking and germination and dormancy, It's really practical information that can be useful if you're testing seed quality and planning germination experiments. If you are looking at seed collection and storage, the Spore Bank guidelines are also a fantastic resource, particularly if you need to buy or source seeds, or you're looking at direct seeding or seed treatments that can improve translocation success. Other ex situ methods that I'll now highlight offer particular advantages, but they do require a higher level of technical skill to get into storage, and it can be time consuming and expensive to develop protocols for each species. They also require more maintenance, which can really multiply labour and resource costs for a project in the long term. You'll likely be familiar with living collections held in botanic gardens and arboreta as part of a species conservation strategy. But this collection type also includes field gene banks, seed production areas or seed orchards, and nursery collections. Plants in living collections can provide material for restoration, translocation, research, education, horticultural development, or display. The emphasis in Chapter 11 is that the purpose and the number of plants in the living collection needs to be considered at the time of establishment and throughout the maintenance of the collection, so we understand the genetic diversity that is captured and available for use. Living collections are really important resources, particularly when access to plants in situ is difficult or further endangers a species, and the Wollemi pine is a good example here. This chapter includes case studies on species such as the King's Lomatia from Tasmania, the Care for the Rare program in Victoria, the orchid germplasm collection at the Royal Botanic Gardens Victoria, which we just got an insight into, and the challenges of bulking seed collections for restoration or conservation. We also have a case study in this chapter from Auckland Botanic Garden on the ex situ management of plants that are highly susceptible to myrtle rust. This Chapter 11 also includes a case study on the critically endangered native guava. This species was common and in good health before myrtle rust was introduced to Australia in 2010, but it has suffered significant declines as a result of this pathogen. Ex situ conservation is needed to ensure native guava will survive while long-term recovery options are pursued. So ANPC has begun a new collaborative project to help establish meta collections at 6 locations in Eastern Australia. For species such as native guava, meta collections provide a duplicate or backup at other locations in a similar way to the backup of the ASBP seed collections at the Millennium Seed Bank or agricultural seed collections at the Svalbard Global Seed Vault. The new project we're starting is a collaboration between botanic gardens and government agencies in Queensland, New South Wales, the ACT and Victoria, and has received funding from the Australian government. So stay tuned to hear more about this project in future. Tissue culture and cryopreservation are additional ex situ conservation methods in our toolbox. The germplasm guidelines includes an overview of the process and a list of the species stored in Australian germplasm banks using these methods. The number of species stored in this way has increased significantly, and a wide range of tissue types can be stored. Cryopreservation is particularly appealing for long-term storage of plant parts, including seeds. But there's plenty of work to do establishing protocols and refining these for different species. So the resource intensity for these methods is high and reliant on skilled staff and access to specialist equipment. In this edition, we've added 2 separate chapters on collection and storage of germplasm for groups with more complex ex situ requirements, including orchids, ferns and bryophytes, and symbionts such as mycorrhizae and rhizobia. Using resources like the germplasm guidelines means we can continue to build on the experience of others who are dealing with similar taxa and germplasm types, rather than trying to start from scratch each time. For each ex situ method, it's really important to consider the end use of the collection and how healthy plants will be regenerated and returned to the landscape. For seeds going into translocation sites, this might mean working on germination techniques and understanding the seedling stage of development and its interaction with soil type and moisture stress. For planting out, you of course need to consider hygiene and site requirements and other factors that go into the translocation planning process and monitoring and follow-up, even if you do have plants in the nursery ready to go. So you can see there are lots of alternatives to translocation, either as a stepping stone into the future or as an additional means of securing and managing plant diversity. Spending some time looking at all our different guidelines will mean you can make the most of the resources available and the time of dedicated staff and volunteers who are involved in your project. I'd particularly like to acknowledge all the authors who contributed to the guidelines, and particularly thank the chapter lead authors and the steering committee members for their hard work and support. And a big thank you to the Ian Potter Foundation for their grant funding to review the guidelines. Thanks so much, everyone.
Thanks, Amelia, and thank you to Maurizio and Peter as well. And thank you to you, whoever's watching, for attending day one of this symposium and for bearing with our few little technical glitches. I thought we'd have more, to be honest, so I'm pretty happy. I hope there are some things you've taken to ruminate on. Please let us know if you have additional questions. We can forward them to the presenters. On the 28th, we have this again, 12:30 to 5. We're going to be talking orchids, mycorrhiza, field monitoring, mistletoe translocations. That sounds kind of cool. Please, if you have anything that you want me to do for next week, you can send me an email or pop my email address in the chat. But also we will be sending out a post attendee survey, so you can give anonymous feedback to tell us how to do it better. Wishing you a great week and weekend. Chris, I'm pretty sure the same link will work again next Thursday. Correct me if I'm wrong.
Yep, that's right.
Yep, same link next Thursday. Okay, everyone, good night. We're bang on 5 o'clock.